foundation to the innovative discussion and interdisciplinary perspective, reflecting the true spirit of global collaboration in healthcare research. So, before we begin, now I would request our distinguished dignitaries, Professor Dr. Sahil Arora, Dr. Jyotish Srivastava, Dr. Rahul Pratap, Dr. Rahul Singh, and Dr. Anand sir to please come up on stage to welcome our speakers of day 2 session of a 7th conference with a planned sampling. I would also request Dr. Ashish to please come up on stage. Now I would welcome Professor Dr. S.K. Meena to please come up on stage. Okay. We have Professor Dr. Ivy Chung here. Please come up on stage. I would request everyone to please give a round of applause. Next, we have Professor Dr. Bhupesh Goel. Okay, Dr. Keshav Raj Ponnel. Next, we have Professor Dr. Tina Chauhan. Professor Dr. Vaibhav Agarwal. Professor Dr. Brian Oliver. Professor Dr. Bhupesh Goel. Thank you so much, dignitaries. Your request to please have a seat. Ladies and gentlemen, we now move into the plenary discussion of day 2 under the overarching conference theme. It is my honor to introduce our esteemed session chair, whose guidance and academic leadership will steer today's plenary session. Our chair for today is Professor Dr. Brian Oliver, distinguished professor. So please come up on stage. Professor Dr. Anand sir, please come up on stage. And Professor Dr. Sahil Arora sir, please come up on stage for the session chair of us. We now move to our first expert talk of session, Professor Dr. Tina Chawla, Principal, Goenka Institute of Physiotherapy. The topic of the session is Innovative Technology Shaping the Future of Physiotherapy. Please come up on stage.
Good morning, everyone. काफी half an hour से हम wait कर रहे थे बस wait कर रहे हैं. So let's not waste too much of time. Good morning, everyone. So as we are bound with the title of this speech, integrating research, technology, and care. So I have brought something like innovative technologies in shaping the future of physiotherapy. The basic fundamental of my speech today is augmenting human intelligence with the intelligent technology and how we are going to do that. What are the developing things are available, how we can execute that, what are the problems will come across, and how we can reshape ourselves with the help of technologies. Okay, so modern physiotherapy right now is going through a lot of transformations, and and even technology, a lot of technology is coming in a market. So how we can integrate AI, robotics, and VR for basically optimal patient outcomes. Because we are dealing with the patient's clinician. As a clinician, we are dealing more of service, treating a patient and all. So the very advantageous thing is that for certain things, like I feel like I'm using this, and next day I feel like it's not helping the patient. Immediately I can stop and I can give a feedback. I don't have to wait for 1 year, 2 years, 5 years, like that. So this is very key feature of our profession. So how this is going to change is like what are the changes, opportunities, and how we can synthesize that is. We'll go into our next slide. So here we are going to change standardized care into precision care. Okay. So there are 4 key factors in this. When you add a tech-enabled transformation, that is precision, engagement, augmentation, and access. Precision means what? Precision means the AI works on 2 factors, that is key capabilities, hyper-accurate movement analysis, and data-driven clinical decision making. The first one suggests that, okay, if someone is walking, okay, my eyes cannot record the degree of deviation. But when it goes to the digital things, it can record a minimum 1 degree of deviation also. So that is a big advantage. And that advantage is how it is useful into a daily clinic basis. That also I'm going to tell you. Second is data-driven clinical deviations, like day-to-day factor changes and how the 1 month, 1 year data can be compiled and can see in one slide. Okay. Augmentation is with the use of robotics and exoskeletons. There are some studies to that. There are many studies. Out of that, I have brought one list. Okay. In that, 3 outcome measure, that is the functional independence and mobility and quality of life. You can see the data almost double it has been augmented. Okay. These are the curve. It is in the flow chart. You can see the below one orange and green mark. devices which are very best for home exercises, and there are some devices which are private release, or you can use it as a scalable. So first one is Fire 3D, which is actually markerless and which is gold standard, means it is moreover used into research basis. It has got a high accuracy. Kinetic tense is also again markerless. It has got advantage of 360-degree capture. The home exercise squad help is an AI app, and because of that, the accuracy is so high that it claims to reduce 70% surgery. AI help is actually you can use to a simple mobile device. So this is also very good. Everybody has your advantage. Third one is Camtai, and my jump tab is like pocket friendly. You can put it into your pocket, and more use in the sports. Where someone is doing a jump, doing and running the heights, speed, like that. Second is wearable tech. This is absolutely like living at the moon. So here we are shifting hardware, software, human input. Okay. So almost 4 types of things are nowadays available. IMU ports, it's a digital goenka which records the very high accuracy, joint range of motions. Smart clothing, where person wear these clothing and it is sensor based. So micro level changes are also monitored. Like at soft stumps, there are some food sources are available. Okay. Another is like wearable biofeedbacks, where someone wears that and if you go out of the range, it will like other with the feedback you are corrected. So a lot of audible cues are available. Next is VR and AR. So here it is like it is using rewiring the brain and body, overcoming the damage pathway and working to the neuroplasticity. So two basic theory, it is mirror therapy, which are actually they are also using in a daily life also now. I mean from long back. But still through the AR, how it is changing. There is one mind-based app which actually works on pain and speed, so it avoids motor and enhances the motor recovery faster. Precision biofeedback, so Numera is like it measures into some millimeter changes. Okay. So why I'm highlighting some key points and what is the effect of that key points into your daily practices. That I'll show into the almost last slide. So basically, it works to the visual motor illusion, that is neuroplasticity. Second, to the AR VR, we can use it for chronic pain management. This is very interesting. How it manages the pain. To the exercise, we are using IoT cans, lot of devices for pain management. But with the exercise, how it is helping? So one is basic fundamental is hijacking the pain matrix, body ownership illusions, and the third is virtue credit exercises. Pain matrix means lot of other cues, for example, other proprioceptive, kinesthetic, that receptors working, touch sensation, to that there is a bombarding of lot of cues. Okay, so that pain is not perceived. Pain is what? It is a perception. Okay, so it is pain is not perceived, and perceived between the movements. Second is body ownership. In that, what happens? A virtual environment will create another me. Okay? has turned into, even our schooling system has turned into a application. Like that, this is where advantage is. Okay? So here patient will be playing like some games, the shoulder range is not there, they just keep slicing fruits. Okay? Second is adaptability, this is a very good feature. Okay? If someone is having difficulty to reach there, okay? Initially they will try to create a more harder one, but once it is perceived that it is very difficult for the patient, so the range is little lower for their reach. Okay, so they are not disappointed. Oh, I'm not able to do that. Third is social competitive environment where the other virtual one will be having a racing games. So it feels very enthusiastic and keep motivating the patient for doing exercises. Because non-compliance is very key feature of physiotherapy. Exercise करना बड़ा बड़ा ही बेकार लगता है, बोरिंग लगता है, क्योंकि रिवॉर्ड में कुछ मिलता नहीं. But gamification you will have 5 reward points. कितना अच्छा लगता है. Next is this is very important for research and all purposes, and even insurance purposes if someone is working outside. So result and data visualization. So here you have a data, okay, which objective data turns into tangible progression and empowers the patient. The patient at the end, patient will feel like, okay, I'm increasing my mobility. I have recovered from this much to this much. But many times patient will feel, okay, why I was suffering? I should have come before also. So the guessing game has turned into visible trajectories where you can see like there are 3-4 plots are available in different ways from starting assessment, progression and end assessment. And the second one is like graded exercises, how much level you have increased. Third, it is used like whether you are ahead of the track, on track, or you are going same as second, how it is result and data visualization is important. It's like functional scoring and engagement matrix. Okay, in that dynamic recovery score is there. Your strength is improved. How much it is improved? It is quantifications. Okay, improve. बहुत अच्छा, बहुत से बहुत ज़्यादा अच्छा. That is not a quantification. You can't use for research purpose. When it's number 1 to 10, 10 to 100, and that way, it's greater impact. So that parameters like strength, mobility, vitals, even cortisol level is also important. So someone is not doing over exercises. Radar charts, there are some components, there's some chart I will show you. Like one patient has improved more on endurance rather than strength or a range of motions. So one can see like, at least I'm not doing this component, but I'm very good in other components. Kinematic here it is pre and post when we have a red map and then turning into a green map, like that, the color coding. Next is neurocognitive priming. Okay, this again works on mirror therapy. It generates the digital thing. Okay, so the kinesiophobia is reduced. Success is well documented. Okay. Subtle game visualization. This I was talking earlier also. This is very important. Okay, कई बार patient हमारा मैं वहाँ बैठकर puzzle लिया था मजा नहीं आया. But when you have a subtle game visualization, okay, even a small degree of after first day 10 degree का, at the end after 10 days you have improved 15 days. So that quantification of 5 degree shows improvement. Patient will feel satisfied for as sparing the time and even cost wise. That way to work like that. Okay. Next is future. Future is synthesis, human empathy plus AI efficiency. This is important. Smarter care. So the hybrid model, you don't have. We are talking about that one cannot have to choose like between the AI and a human because human cannot. AI cannot give you empathy factors, emotional factors, support like that. So integration is very important. So hybrid model where you have an AI triage, expert daily diagnosis, technology, which turns into a scalability and a best outcome measures.
so much, ma'am, for your nice insights on the technology and AI. But what I feel, as you have mentioned in the last slide, that we are lacking in infrastructures, right? Because this type of technologies and AI things are only possible in multi-speciality metro cities, right? Thank you so much.
Thank you very much. I think in Australia we live with this idea that clinicians are frightened of AI because there's a fear that AI could take over their jobs. But do you have any perception how physiotherapists in India would perceive that? What happens if patients don't want that? If patients don't want to talk to someone, so when people are busy, if you can do something online and not have to wait to see someone, not need an appointment, there's a lot of advantage. So the patient may opt out of what you think is important. So that's the thing from my perspective. Just a suggestion for you. If you want to do this, work with sports scientists because they've been doing it for years. They have the technology, they know how to use it. They don't do it from a rehabilitation point of view. Yeah, but they basically have improved in performance, but you can use their technology and enhance your practice.
Professor, that's a really nice information which you got today. My point is that as technology is growing, are we modifying our syllabi in such a way that yes, now rather than looking into the patients, doing some exercises with the patient, searching on that particular part, are we using these AI robotics and technologies for our research with our students? Or we are teaching them how to make new softwares within physiotherapy, because it is not only the task of yes, that's right. If it is adding up, yes, students in future will be able to make softwares, not only the clinicians. Any update from your universities? Any software which has come out? That's what I would like to know. I think the students would like to know.
Currently, we are working on a teaching interface model from AI perspective. There is a technique in physiotherapy called myofascial release. So, it's a hands-on therapy, which is so precisely we need to be very accurate in applying 1 milligram force to 5 milligram force. So, we need to quantify that. And plus, there are technical differences between compressive forces versus shear forces. So, by educating candidates, we need to make it outcome measure or feedback kind of mechanism. So we are working on a sensory cloud, which is definitely helping us to feedback. So this is first phase of building.
Thank you very much. Thank you.
practice.
So we are reading so many books where we look at how to eliminate pain. Sometimes we hit the thoughts, other times what we do is like fool the brain that pain is not there. So that fooling happens. So you're like initiating a movement, sometimes restriction is not because of some issues, it's just because of some fear. So that psychological pain that can be indicated into avoiding other engaging the sensory feedback and so that pain is eliminated. See, pain again is very vast topic depending on the person. It involves, for example, if there is a big injury, that means there's a where we need nothing. So it has to heal. You don't have to do anything, just to regenerate it. Okay? If you want to enhance, you can use some healing modality. But when pain is psychological, some restrictions are there because of muscle spasm. So in that case, that agitating breathing mechanism is doing.
Thank you, ma'am, for presenting a futuristic version of physiotherapy practice. Your session was truly inspiring and innovative, driven thinking among the audience. I will request our session chair and esteemed dignitaries on the dais to present a token of appreciation to our speaker, Professor Dr. Tina Chaudhary. Please give a round of applause. Thank you so much, ma'am. We now come to the most prestigious segment of the morning, the keynote address, which sets the academic tone for the entire day. It gives us immense pleasure to introduce our keynote speaker, Professor Dr. I.V. Chung, Professor, Department of Pharmacology, Faculty of Medicine, University of Malaya, Kuala Lumpur, Malaysia. The title for today is Translating Mechanistic Studies into Affordable Therapeutics for Cancer.
Hello, good morning, everyone. It's really a pleasure to be here. Thank you so much, organizing committee, to really host this talk and invite me over to this good conference I heard since yesterday. So my name is Ivy Chung. I'm from UC Malaysia. How many of you have been in Malaysia? No? Or just one? So I invite everyone here to, if you come to southern of India, please visit us in Malaysia. And if you are in doing research, you can come to my university. So yeah, so I've been working in the laboratory. So I'm trained pharmacologist in the laboratory for most of my career. So I'm going to talk a bit what I've done in the past 15 years, picking on one specific topic to share today, and then also try to share with you what's the next direction in my second half of my career, and how we can, from a laboratory scientist like myself, to move on in the research career. So this is what my talk today. So I think for those who are in cancer therapy or in cancer research, we have been working very hard to cure cancer, right? So a lot of effort has been done to focus on tumor cells, right? We try to get drugs, novel drugs that can target whatever cancer cells you have in any parts of the body. But my focus from beginning, even from starting from my PhD, I've been focusing on the host cells. So in our field, we call it tumor microenvironment. So I think for those who, I'm not sure what's your background, just give some introduction. Cancer cells grow, they're all coming from a normal cells that go haywire, they're just mutated. But they don't grow alone. They need a lot of so-called supporters within our body to help them to grow and progress. So this is the environmental. So for example, in this slide you can see, we have tumor cells, the one here, the really big pinkish cells, but you also have others, different color of cells, different shapes. You can see with all the labeling there. They are all the tumor microenvironment, the host cells. So I've done a lot from endothelial cells in my PhD. Then when I joined as a faculty, I work a lot on CAFs. CAF is the cancer-associated fibroblast. So they are not tumor cells, they are fibroblasts, but within the environment of tumor. So we call it CAFs. So this is the main topic of today. But recently, our group also has ventured into macrophages and also neutrophils. I hope I can expand more in the next few projects in the future. So this is the main topic of my talk. And to start, we've done a lot of CAFs in multiple cancer models, but I think for today's presentation, I'll focus on endometrial cancer, because this is the cancer that I worked for the longest time, starting after my postdoc and work in academia. So just a brief introduction. So endometrial cancer, also known as uterine cancer, maybe that's more familiar within India. That is the second most common gynecologist worldwide. I'm not sure how many cases in India, but I'm sure there is some, right? And actually in developed countries, this is where it's a major problem. And there are two types of endometrial cancer, type 1 and type 2, very easy. But basically most of us, or 90%, 80% is mainly type 1. It's associated with age, it's associated with estrogen or BMI, right? And type 2 is estrogen independent. So from biology's point of view, we will always look at disease based on how they occur. So estrogen dependent or independent really makes a large difference of what kind of endometrial cancer you have. And as mentioned, most of the type 1 endometrial cancer affects people older, 60 years and above. These are the 60 years is postmenopausal. That means they are already into menopause, so there's no regulation of estrogen, less estrogen regulation of estrogen, hence type 1 arise. But I think the increasing data in worldwide showing that there are more and more women getting endometrial cancer younger. So even under 40, 60 to 40 is huge gap. And why is that, right? Endometrial cancer, we thought it is okay, it's an old age disease. It happens when women already past the so-called childbearing age, but this is no longer the case. So this is really a problem worldwide to figure out how can we deal with endometrial cancer patients. And one of the single biggest risks that is associated with EC is obesity. So it's something that I think is important to take note. But I think that half audiences are women, and also you have family, you have mothers and sisters, and also your daughters. I think it's important to keep our BMI in check, right? Not just for beauty, but I think it's also important to keep for health. So there is a lot of evidence showing that many, but done by multiple countries, just obesity alone can give you about up to 5 fold increase of risk of developing endometrial cancer. And there are a lot of biologists discussion behind that. But one of the important things why this matter is that actually endometrial cancer is easy to manage. You just take out the uterus. Now for women below 40, this is a problem. We still want to get children, right? So how can we do that? This will be a real dilemma. If you know Angelina Jolie, she's the one that took out the whole thing. She's done with it. She took out the whole thing, but she's a different case. She's a BRCA1 mutations. So for those who are under 40, we have good treatment. It's very, very manageable. We don't use chemotherapy, we just use hormonal therapy to manage these young patients with EC. But because of obesity, this hormonal therapy, or we call it using MPA, it doesn't work as much. So from 90% down to maybe 30%, right? So that has shown that if you're obese, your chance of benefiting from hormonal therapy is way down compared to the non-obese patients. So this is where scientists like us will think, why is that? How can we understand better so we can better treat those women who are obese but having endometrial cancer? But remember, they also get endometrial cancer because they're obese. So this is where the interplay between the host environment, the host physiology with the disease. So here's where I come in with calf story, right? So on the diagram on the right hand side is a hallmark of cancers. If you're in cancer research, you'll be seeing this for more than 15 years. First started with so these hallmark cancer are the characteristic of cancer cells. But here I'm showing you that calf or cancer-associated fibroblasts also contribute and show similar hallmarks. So they are no longer like acting like a very, what do you call it, a very passive actor in the neighborhood, but they're actually very actively contributing like tumor cells. So the first set of the work that I want to share with you is from my first PhD student, Dr. Kavita. So what she has done to answer this question is, if you want to study calf, we have to figure out where to get the calf. Even today, it's very difficult to purchase cancer-associated fibroblasts from endometrial tissues from commercial companies. Very difficult. The only way to do is to collaborate with the clinician when they actually do operations on women who have endometrial cancer. We get some tissue and we produce our own cells. So this is called primary cells. So she has isolated calf from various endometrial tissues. Here I'm showing 4 different patients, but actually we did more than, I think she did more than 30, right? Just to see whether we have the right biology. So every tissue may show a bit slightly different response because of heterogeneity. Everybody is different. But what we found, regardless of what patient samples we use, the products from the fibroblasts, so fibroblasts secrete a lot of, they secrete stuff. They like to secrete stuff. Their job is to secrete stuff because secretion. Their secretion, when you lose the secretion and put back to the tumor cells, ECC1 and also HEC1A, they are all EC tumor cell lines. These are cell lines. You can see with the calf, with open label, right? The tumor cell actually grow in response to calf secretion. So calf is doing something to the tumor cells to grow back faster. But when we compare with another type of cells called THESC, THESC is our so-called benign or normal endometrial fibroblast control. We use it as a control. They are from the normal endometrial environment. They are not related to tumor cells. So when we use that and test in the same experiment, we see opposite, right? They're actually killing the tumor cells. So what this means is very important. There's a very important discovery that we still don't know how to do this. If in women we have fibroblasts in endometrium, you have small budding tumor cells. Actually, if your fibroblast in your endometrium is benign, they are doing something to keep the tumor cells suppressed, not growing. But when the tumor cells really grow up away, the benign fibroblasts become calf, then the fibroblasts now turn their face and then promote the tumor cells. So they change behavior. And that kind of physiological change is, for us, is very interesting because we want to know how they convert. But this is actually a study for future to know what makes them change. Now, we also figure out what actually that the calf secreted to influence the tumor cells, right? So we've done a lot of proteomics work or ELISAs. This is the final result. We found that interleukin-6 is a cytokines. They are overwhelmingly produced by calf, but not in the benign fibroblasts. And with that, we also have identified that the interaction of the calf secreting the interleukin-6, the IL-6, is one-way communications. Because from here, If you know, you've done some research in the lab, it's called Western blot. We measure the protein. So protein analysis of interleukin 6 receptor and also GP130, these are the receptors for interleukin cytokines. They are only expressed in the tumor cells. So ECC1, HET1A, all these first four bars, they are tumor cells. And the last four, they are with the FIP at the end, is fibroblasts. So calf secrete IL-6 for only one reason, for their tumor cells to use them. So what happens when interleukin 6 go and bind to the receptor in the tumor cells? So we figure out that upon binding of interleukin 6 to the receptor, we call this a signaling cascade. So there's like a wiring network within the tumor cells. This may take a while to understand, but I can just tell you the outcome. So basically, when you bind IL-6 to receptor, you just trigger a lot of cascade of signalings, phosphorylations. You hear about protein like STAT3 going into the nucleus and then produce another protein called c-MYC. c-MYC is a transcription factor, if you know, that can generate a lot of expression of different kind of so-called aggressive proteins that can make tumor cells happy and grow and disseminate. So this is what the function of c-MYC. And when we look at the so what happens is that when we look at the we treat the tumor cells in the presence of AD412, is an inhibitor for interleukin 6 receptor, or we treat with an inhibitor for STAT3, then we realize the cuff effect no longer work in tumor cells. So it really shows that this pathway is the dominant pathway, how the cuff talks to the tumor cells. So that's like I'm talking to you now, right? You hear it because I have a mic. So, but if you take away the mic, you relax. The same thing. So we want to identify what makes you get communications. You just break the barrier, you get less. So tumor cells will not have the influence by cuff, and they may stop growing or grow less. So this is the so-called paradigm of thinking, how we want to understand, why we need to understand the tumor microenvironment. Now back to c-MYC, right? The c-MYC is the ultimate protein. So I will skip some of this. Basically, what happens is we generate the tumor cells without c-MYC, so we can just knock out the gene in the tumor cells. And when you do that, we put that back into the animals. So let me show you the graph. So what happened is when we implanted tumor cells alone, the original one, they call it ECC1, they grow like this over the about 6 weeks, right? When you put calf alone in another set of animals, they didn't grow because calf is not tumor cell, they will not grow in the mice. You add these two together in a separate group, right? You can see how calf, in the presence of calf, the tumor cells grow faster, right? It's growing faster than tumor cells alone. Now, when we injected the ECC1 without any c-MYC expression, which means they can't express c-MYC, on their own, they grow less. So c-MYC has a real function to tumor cell biology on its own. What happens if we combine that with the calf? So you can see from the red color, compare the red color with the purple color, there's a big difference, right? So calf initially can increase the tumor cells here, but now with c-MYC knocked out, it stays like itself, nothing changed. So this is a very important observation that we have seen in the animals, that it works in the animal too, right? c-MYC is the key. So of course, with that, the idea is that can we do c-MYC inhibitor and treat patients with that colorectal cancer? We can, but of course in the lab we're more curious about the biology. There's another student here trying to look at if calf can make tumor cells to grow faster, can they make the tumor cells to move faster, right? Cancer actually can be treated if they don't move. They stay in your body, they don't move, we can do something. The surgeon will say, I can take it out, no problem. But we need oncologists because why? The tumor cells migrate. They spread to every part of the body. That's very difficult to do surgery. Imagine if you go to the lung, how many punctures you can do in your lung, right? To take out tumors. So they do chemotherapy or they use pharmacological intervention to go systemic intervention. So this my next student asked the question, how to see their interactions. So what he did is that he labeled the tumor cells the tumor cells in red, and then the calf or the benign fibroblast has a control in green. And then he just mixed tumor cells and one of the fibroblasts in the same dish, in the matrix gel, to see can we capture what happened. So here you can see that if you are culturing the tumor cells alone, they grow like a little ball, like a ping pong ball, pop, just like that, with no structures, very loaded, very heavy. If you grow the calf alone, they don't grow, it's green, right? You don't see much because they don't spread, they just spread themselves, they don't leave, they don't die, but they just stay there, they don't really grow because they are not supposed to grow like tumor cells. They are calf, they are fibroblasts. When you add them together, you see how the biology has changed. You can see there's a lumen, right, in the between, and you see the red color is all like a perforated, bigger, the size bigger, and it's perforated. So something's in the middle. So what happened is that we did a live imaging over 24 hours. We can see, right, the red color dots are tumor cells. They just grow in the first 0 hour to 24 hours. They're just like that, growing. But for the calf alone, green color, they just grow, but they have the kind of starting to have interactions. They interact with each other. So fibroblasts interact fibroblasts like holding hands. They're trying to search, having network. But when you put this together over the 24 hours, 24 hours alone, you can see that the coordination of the networking is even more prominent. The calf actually make better hand holding, and there are tumor cells related in the red color. On top, actually, they can, if it's a video, you see rolling down the hands of the fibroblasts. So it just gives us an idea, perhaps the fibroblast function here is to make the pathway, make the road, so the tumor cells can walk onto them, right, and meet each other to make the sphere. So that phenomena, it only happens if you culture the tumor cells with calf, but not with the control fibroblast. This is the one on the right. Nothing happens. So it's very specific to calf. I think we also, through this experiment, we also showed that the calf can increase the EC motility because we can measure the speed they move, right? Tumor cells motility increase significantly with in the presence of calf, but not the other way, which means tumor cells do not affect the migration rate of the fibroblast. So it's only one way. And the next, another student was trying to understand because she said, yeah, we know how calf responds to the tumor cells, but what does it do to the hormone, the MPA, or the hormone that we use to treat endometrial cancer? We know MPA works, but can, because of calf, affect the patient's response to the hormones. So she asked this question by making a rather complicated tumor models. So it's very difficult for us to get calf already, right? To get patient and do that. Now she even have to get more so that she classify the calf into two groups. Calf that isolated from patients who are obese. So we check the BMI. If they're obese, their calf is called obese calf. If the patients are not obese, that means BMI less than 25, we follow aging standard, then it is non-obese calf. So she measured she grouped the two calves to study. So at the first glance, when she looked at the cell cycle regulator, stromal differentiation, which basically it shows the performance of the calf, right? You can see some big differences. On the top panel, these are any calf from non-obese, they are all lean calves. There's not much red, but there's some certain rates coming up, right, in the obese calf. So you may say there's not much difference, right? Dr. Ivy, there's not much, what's going on? But in terms of fibroblasts, they're not supposed to change much. But with this small change in the stromal differentiation of cell cycle regulators, it makes a difference in the biology. I can show you that later, right? And this is it shows that because of you are from obese, with the MPA response, you actually proliferate higher. So BRDO is a labeling we use to show the nucleus actually dividing, right? So you see from 10, let's say in the presence of 10 nanomolar of MPA in the presence of hormone, it was here for if you are not obese, but let's say you are from obese fibroblast, it goes up quite a bit. So this is the effect you see. Now, going back, you understand how that happens, right? Hormonal therapy is supposed to suppress your endometrium, but in this case, it actually improve or increase the proliferation. So at this point, she focused on the effect of MPA into the cell. So we focus on the metabolism. Here, there's a glucose metabolism pathway. So MPA actually directly affects RRS2 signalings, and then that's how they come into the Krebs cycle, also with the glucose pathway. When we measure these highlighted enzymes, right, it shows, you can see, here is the from the CN, non-obese, here is the obese. It's just red. It's a red C. Although there's some reaction, we expected some overexpression, but we don't expect so much if you are from the obese calf, right? And that actually, so we measure the GAP DH activity, it actually shows this increase. So here, transcriptional analysis is only at transcription level. It doesn't mean a lot until we see a functional. So GAP DH activity here actually shows that it's actually turned into a functional product of the glucose metabolic activations. And then when we want to test, is it really true, for real, we are pharmacologists, we use a lot of chemicals. So we test one with this IGF-1R receptor, right? We block this, then we can see a reduction, a reverse of the activation of GLUT6, GAPDH, and also CD36, right? And that translates to less proliferation by MPA in this obese calf. And we also do another one, we block MPA receptor. So make sure if I block MPA receptor, then does it also say, observe the same thing? We observe the same thing as well. So it's really tied up how MPA leads to, MPA activations leads to IGF-1R receptor. They can be the channel of how the interaction between MPA in obese calf. I think this is the key, the message that we got from these experiments. Now, then another student tried to say, yeah, we talk about glucose, but we know, right, obesity is not just glucose, it's not a lot of sugar, but a lot of lipids as well, a lot of fatty acids. So the other right-hand side of the same pathway I've shown you, the green color, these are from the, and look at the enzymes related to fatty acid synthesis or metabolisms, right? You do a similar kind of concept. Now, this student's very ambitious. She said, I need more calf for obese. So she collected more, and they can see from this set, right, this lipid fatty acid enzymes sing up and down is a base, but you can see from here and it's a lot more red and very selective. So you may say it's very heterogeneous, right? The response. It's true, remember, each column is actually from each different patients. Different patients will eat different things in different physiology. There's meant to be very variant responses, but it's the overall that we're looking for. So this is what we feel that is really striking evidence to show there's abnormal lipid metabolism if we are obese, right? Okay, then now this is very interesting. So if you're obese, then what happens? Actually, in our cells, if we have a lot of fatty acids, you don't use it as energy, you get stored. Like in body, you see a lot of fat in your belly, right? Belly fats. But in cells, actually, they will store too. So this is a staining we use to called ORO staining to see the lipid droplets in the cells. The more red means more lipid in the cells. You can appreciate here when you treat the calf, whether obese or not obese, they are just a lot more reddish after treatment. So hormones actually cause you to get more fat. This is the results. We quantify that, that's fine. So if that is the case, then can we reverse, right? We know that these fatty acids actually will pass through this PPA-alpha signaling. There's a long story, it's not, I didn't identify that, it's a lot of literature shows that it's through PPA-alpha, and that's how we can control those metabolic genes that I've shown you before. And we postulate that this activation leads to MPA resistance. So we use an inhibitor, PPA-alpha inhibitor called DW6471 to inhibit this, and then to see whether is it true. So indeed, here I just go through all the things to show that if you inhibit the cells with this inhibitor, it totally reverses the upregulation of these genes by MPA in obese calf, right? And that one of the proteins that we look specifically is FASN. We are interested in this because we find that these genes, it's called fatty acid synthesis, enzyme that produce more lipid. So if hormone treatment can make you have more lipid, they must be through a synthesis, so a de novo synthesis. So that is really shows that inhibition of PPAR alpha can stop that. And that could give us an idea, can we use PPAR alpha inhibitor in our clinic? So these are the few things that we do in the laboratory to find which target, and then how can we modulate that pathway to reverse the phenotype or the disease conditions. So we have done also in the same studies we use with the inhibition, the lipid droplet reduced significantly, right? So what I told you is, if you're obese, PPAR alpha is activated, you got lipogenic phenotypes in the calf, and calf will strip more lipids and more metabolic crosstalks to the tumor cells. But we want to know, in that case, would the tumor cell grow better or grow slower with the inhibition? So here is a story where if the tumor cells are treated with the calf from obese, they grow faster, right? And then if you treat with MPA, even more. If you block the MPA actions through GW6471, it reverses. And yeah, it's a lot more, but I want to summarize here just to have a principle that you can bring home what I'm talking today. So here are two mirroring situations. Both are ladies, only ladies have endometrial cancer. The difference is that whether you are skinny or you are big, heavy, right? So on the left, if you're not obese, calf, you get MPA treatment, you get signaling. You have the calf will still talk to the tumor cells, provide fatty acid source to the endometrial cancer, right? But the calf is less lipogenic, right? Therefore, less lipogenic will be less promotion of tumor cell proliferation. And because of that, we think the tumor cells are more sensitive to the hormonal treatment. Contrary, if you are obese, right, just the person is obese, you have endometrial cancer, your calf will be obese, more lipogenic, yes, then will promote more fatty acid sources and mediators to the tumor cells will grow faster, and that will also reduce the MPA sensitivity. So this is one way of one perception, that one way of perspective we have, that when we treat patients, we don't just say you have disease, but we need to know what kind of body you have. So it's called the host and the environment. So this is our current hypothesis. I think because of time I can just skip through, but I want to bring you to the next two more minutes about what we are doing next. So I've been doing that kind of work, animal laboratory for some years. But I think working forward, I think it's important for you, especially the younger scientists, to think whatever we do, we must make sure it's translationable, impactful. So do as much as you can in the beginning to learn the skills. After a point, you must know how to translate that. So recently, I have been have a great opportunity to work with a bigger group to form this iData based in Malaysia. So here we are trying to drive healthcare solutions through partnerships. So what does it do is that in the using layer, I lead this program. It's an academic platform that we can do collaborations among parties. So we do collaboration with academia all the time. We do some collaboration in industry. But to develop really good, I would say, effective healthcare that can reach patients, we need more than that. So we decided to bring into the government agencies, the regulatory bodies, as well as the patient advocacy. At the end of the day, we work for who? Not for government. We don't work for university. We work for the patients. And who are the patients? It's us. We could be the patients. Our family members, our friends, people that you know. So I think having that mindset from beginning is key, because this will take a lot of growth in yourself, how to deal with this kind of collaboration. No longer it's about your skills, your intelligence, but it's also your EQ, right? Your openness to collaborate. So here we are working at international levels. We also in the region, because we know that in Malaysia or in my university, I can't deliver this alone, right? We have to work in multiple countries to bring the best solution. Best may not be the most expensive, and this is effective, backed by good science to the market, to patients. So here we're proposing doing drug repurposing. Now, why drug repurposing? Because a lot of times we do in the academia, we want novel, right? We want pattern. but we don't have the means and the finance to bring a molecule to the clinic. It took so long, took so much effort, finance, and I'm not a billionaire, so I can't fund my own research. But there are patients waiting outside. So what we want to do is there are a lot of this is a database. It's done by the UK Anti-Cancer Fund. They've done this for I think at least 15 years. They have curated the molecules, published molecules in the literature. You can see from here how many numbers of them. We think there is approved by FDA, already in the WHO EML, have some in vitro cell evidence, some may have in vivo, some may even have gone to trials in human, tests in human. So with this, can we make some of these use repurpose it for cancer? They are listed for various reasons, for like aspirin, paracetamol, whatever you choose, right? They already we use it daily that you can get over the counter in the pharmacy. Can we reuse that if we can show there are evidence that can target cancer cells? So this is what the drug repurposing means. So right now, we are focusing on a compound called artesunate. It's a drug that used for more than 20 years to treat malaria, very effective. Malaria is often, I'm not sure in India you have malaria, still have malaria, I think there are some cases. We use this, even in children, it's effective. So we are now doing trials to use artesunate to treat colon cancer and also cervical cancer. Now why is this? It's only cost one pill, 1 day treatment, only $1 compared to, right, and it's already, we don't have to come to hospital, you can do at home. So I think this is the reason we choose these drugs, and we have multiple other projects related to artesunate to understand mechanism studies, how they will work in different kind of cancers that matters to Malaysia, maybe matters to India as well. So basically I just want to tell you the kind of thinking process, how I move my research. So this work is not done by me or by the technical students, or also research assistants, the staff in the hospital, in the universities. We can't work together. It's always collaborative partners that shape our thinking process. Patients are the most contributor to our work, and also funders. So with that, if you are come to Malaysia, just drop me an email. My email is there. We welcome you to visit the lab and also have engagements. Thank you so much.
Thank you, ma'am. Do you see the scarf targeted therapy as valuable in preventing the metastatic cancer in other conditions also, other than endometrial cancer?
Yes, cancer cuff research has been done in a lot of other cancer types: breast cancer, colon cancer, prostate cancer, lung cancer. I think a lot of people do cuffs for a while now.
And it goes in the same way for the OBs and non-OBs.
They may not do so much of that. So yeah, I think no, actually I don't find I have not found any literature that really does obesity. We chose that because EC for endometrial cancer, obesity is the key. So independent risk factor for the cancer. So that's why we thought there's a direct link.
So it is specific for the endometrial.
But it has been shown for other cancers too, for obesity.
Thank you.
Yeah, I think it's hormonal regulations, the pro-inflammatory conditions in the body, long time incubation of pro-inflammatory conditions in the women to promote endometrial cancer.
More prevalent in OBs, right?
Second.
Which is more prevalent in OBs.
Yes.
So this is the reason why this is endometrial cancer, right? In OBs patients.
Yes. Yes.
How interleukin 6 and endometriosis or endometrial cancer.
How does interleukin 6 with endometriosis?
How they are interfering each other, the fibroblastic activity and IL-6, because IL-6 is entire different thing, so you are correlating both of the things in your speech. So how they are associated?
So calf secret interleukin 6. We often think, in other tumor cancer studies, like pancreatic cancer, lung cancer, they secret interleukin 6 themselves. It's autocrine and paracrine, right? Both ways and autocrine. But for endometrial cancer, it's only one way. They don't use it for themselves. They secret, it's mainly for tumor cell. And benign fibroblasts don't secret IL-6. So the calf somehow being influenced in the environment of tumor to secrete interleukin 6 for tumor cells.
But it is very specific with the uterine cancers.
For in my laboratory, that's what we see in the endometrial cancer. But I cannot say for sure for other type cancers.
Because the thing is that if you are dealing with obesity, the papers are coming out in the movement science. Obesity basically promoting the fibroblastic activity automatically.
Yes. That, of course, that is, yeah.
They are automatically promoting. So directly, indirectly, if the obesity is there, then it will be promoting the cancerous things. But you have to look at the IL-6 associated with or not.
So we have another study that is ongoing. So we try to create the obese condition in the endometrial cancer and calf. Interleukin 6 is one of the product that are regulated, but it may not be the main driver. It's still ongoing. So I don't think interleukin 6 is the only story between the obesity and endometrial cancer. Yes. Yeah, sure.
Yeah, actually, interleukins is basically anti-inflammatory and anti-coagulant. And that
so there is also the people shorting it out. If the obesity is there, then the inflammation will raise automatically in the bodies. So I want to just understanding the interacting physiology between the IL-6 and fibroblastic activities between human brains.
Okay.
Both factors.
So in my settings, the fibroblasts do not express interleukin 6 receptor. So in that regard, we believe there is no direct influence of interleukin 6 on the calf. Yes. Because there's no receptor at all.
Maybe I can help.
Yeah, yes.
Maybe I can help out here.
Yeah.
So, if you have obesity, you have increased free fatty acids in your bloodstream. They feed into the arachidonic acid pathway, and that will cause a whole range of leukotrienes, prostaglandins, all inflammatory mediators. Now, they're all context dependent, so prostaglandins can be pro-inflammatory, anti-inflammatory, leukotrienes, same, and it's all about the situation in the body. So in obesity, you have lots of pro-inflammatory, pro-inflammation things going on. And in IV cells, when they get fats, if you make them lose that fat is actually metabolized principally through the arachidonic acid pathway. And the reason I'm sort of keep harping on about that is because I mentioned the NSAIDs. So if you were to use something like paracetamol, which blocks that pathway, then you get shunting between what type of products you get, and you get different types of inflammation. So that connection between inflammation, tumorigenesis, and tumor biology is really complex. It's not just as simple. And I know in your studies, Ivy, you said that IL-6 signaling is one way, but GP130 exists as a soluble thing, and you can actually get trans-signaling back. So if you've got GP80 on certain cells, so we've shown in the airway very similar things, but if you actually have a soluble receptor, you can actually get signaling of IL-6 back on the fibroblasts, even though in the absence of that you don't see it. That sort of signaling is always complex and never requires something to shut it. So I think basically everybody's correct. It's just the viewpoint that you can't run for. It's always complex. So if you speak to people that work in, say, food science, they'll say something like prostaglandins are bad. And they're not. They're actually not good. So if you have a viral infection, prostaglandins go, they open up your airways, it's not starting. If you don't have prostaglandins, you're all dead. You know, it's always context dependent, it's always understanding the system. But the thing is, everything is rats, and that's what makes it so difficult from a biological perspective. So it's sort of seeing the big picture and then focusing back down. But perspective, you have to focus back out. But this is your talk on mind, Mark, I'm quiet, so I will ask you a question. So, in your drug repurposing enterprise, is it possible to patent things? I know you can't from the database, because that's not obvious, but if you were to discover something, could you have a patent and could you push it forward?
Yeah, that's a very, very good question. Thanks, Brian, for helping to answer. I hope that answers some of your curiosity. But back to your questions, yeah, there won't be any novelty on the molecule itself unless there's some interventions on delivery, method of delivery. So maybe formulations or whatnot. For example, right now we are using artesunate. It already exists in the oral formulations. So about how effectively we develop it more targeted the tumor cells receptor, maybe that is where the novelty comes in, that's where the pattern comes in. But ultimately, when we try to select which drug to use, it's not from novelty point of view, it's basically how fast we can make it approved by trials and be used by patients. So it's slightly different thinking process.
I knew the answer, just I wanted to use it today for students.
It's a very good professor there.
When they're off patent, so when they become generic, if you find a new use, you can repatent them and make money.
Yes.
And if you reformulate, you make money. Or if you have a new combination, you can make money. So you don't have to have a new chemical entity all the time. I think that's really important. The ideas are much more humanistic than the issues actually want to see the patients out and make money.
Well, we are looking for fighters in the crowd, that's why. Thank you so much.
If you don't mind, I just wanted my understanding and answer if you can do it, because I think you have a purpose to sorting out my question. There is a very rough statement, especially now in the cancer dissociation medicines. All hormones are proteins of the hormones. So the inflammation process, which was the marker, I am not into the markers. Right now, protein dissociation is a both way. It is not a unilateral way now. According to the new research that I have seen since last year. So is it true or not?
I think if you're old like me, what you find is that the concepts go around in circles. And what you think is new isn't actually new, someone showed you 20 years ago. And I think when we look at biology, that's exactly the same thing. So we think that things are new, but they're not new. We think that things are only going in one direction and go forward. And you actually find that the old papers actually have the answer there. So I think you're right that everything is going on. It's just how we think about it. But in terms of, so from my perspective, in terms of lung cancer, we know a lot, but yet we know nothing. We still can't cure it really. And this is true for most cancers. So the best thing to do is to get the chop it out. But when it comes to chemotherapy, it's very difficult. But the trouble is that cancer doesn't stay the same. So you treat a tumour with chemotherapy, and the tumour changes. And that's where the complexity comes in, because somebody says, oh, this drug works here, and it probably does. But then if you treat with that drug, then it becomes resistant, and then what do you do? And the cells are actually changing, and in the tumour it's not one cell, it's not homogeneous, it's heterogeneous. So you get differential population, differential growth, and that's what makes it always so complex.
I think in my best view of the medical science, the pharmacology people know the depth people of inflammation except that anyone in this universe. But why people are not conjuring and giving an exact answer to us people, and we are just led behind this all the shits of the inflammation story, because you people know each and everything about inflammation more than anyone.
I'll just take it around a different way. You mentioned Chinese medicine, I observed that yes, the concept which was brought in by the Western countries in our country, of course, that yes, a single medicine will be using for almost every patient. It's not acceptable now, even for Western scientists. everybody is now accepting it. Individual patient requires individual, correct? Maybe individual drug will not be able to work, aspirin or anything, if I mention it. Combo, what Indian system or Chinese system used to do it earlier, that will be the beneficial because it's a balancing of hormones and enzymes within the body system. Which has to be taken. I feel that I won't take further.
He's asking me to answer you when you speak Hindi. So basically the problem is that inflammation is both good and bad. We need inflammation, without inflammation we don't. And then when we look at single factors, they work on multiple receptors and do multiple things. So in the context of one experiment you get this answer, another experiment you get another one. And so IL-6 for example, that's my favourite cytokine, it's unfortunate. That was actually discovered by two groups simultaneously. One group thought it was an antiviral, thought it was an interferon, the other group thought it was a pro-inflammatory factor. And it's a really acute phase protein, it's produced quickly, and it does everything. You look at someone the wrong way, it's up in schizophrenia, it's up in obesity, it's up in every disease state. And the truth of the matter is, you've got no idea what it does. It does everything. But how can something that does everything be so important? And yet, if you come into a patient and you get rid of IL-6, most of the time it doesn't do very much. Too much feedback.
Thank you so much, ma'am. Thank you very much, Professor Dr. Arjun, for an insightful, inspiration and intellectual enriched keynote address. Now I would request our dignitaries and session chair to give a token of appreciation to a keynote speaker, Professor Dr. Arjun. Please give a round of applause. Expert talk series of the day, I would call upon on stage Professor Dr. Pallab Bhattacharya, Principal, Gitanjali College of Physiotherapy, Gitanjali University of Airport, to please come up on stage as a session chair. Our expert speaker is a distinguished academician and clinician in the field of physiotherapy, Professor Dr. Bhupesh Goyal, Head, Department of Physiotherapy, Goenka Global University, Jaipur. The topic of the expert talk is Rehabilitation Nutrition Fueling Physiotherapy Success in Musculoskeletal Recovery. Now I request Professor Dr. Bhupesh Goyal to kindly take the podium and enlighten with his expert talk.
Hello, I'm audible? Yes. यहाँ इतने ब्रेनस्टॉर्मिंग सेशंस हो जाते हैं ना कि बेचारे बच्चे जो हमारे स्टूडेंट्स ये सोचते रह जाते हैं कि हमको अटेंड करना है नहीं करना है। और वैसे भी मैं शायद लंच और इनके सेशन के बीच में खड़ा हूँ बच्चों को, तो ऐसे ही भूखी नजरों से देखेंगे मुझे। आई एम सॉरी फॉर दैट, लेकिन आई ट्राई दिस मेक दिस सेशन इंटरैक्टिव। तो हमारा अभी तक का जो सबसे ज़्यादा नेग्लेक्टेड पार्ट जो रहा है वो रहा है न्यूट्रिशन का। कंफर्टेबल इन इंग्लिश एंड हिंदी बोथ, राइट? सो हमेशा हमारा जो नेग्लेक्टेड पार्ट रहा है वो रहा है न्यूट्रिशन का। बीइंग अ फिजियोथेरेपिस्ट हम ये सोचते हैं कि कहीं ना कहीं जो है वो सिर्फ हमारा रोल है किस पे, मस्कुलोस्केलेटल जो हम एक्सरसाइजेस करवाते हैं, मोडालिटीज हैं, इन्हीं सब चीज़ों पे। लेकिन हम भूल जाते हैं कि हम फिजियोलॉजी चैप्टर, फिजियोलॉजी का इतना बड़ा बुक पढ़ते हैं, और वो फिजियोलॉजी क्या है? What is this physiology? Anybody here? I request the organizers to give the mic to the students if possible. यहाँ फर्स्ट ईयर से कितने आए हैं? How many from first year? Raise your hands. Oh, that's great. Second year, third year, final year. Second year, third year, final year. Final year से कोई भी नहीं होते. PGs? How many PGs? Oh, that's great. मेरा जो टॉक है ना वो ज़्यादा उस पर फोकस होगा कि हम पैसा कैसे बनाएं, हाउ टू मेक मनी. ओके, हम सब क्लिनिकल काम करेंगे, वी हैव टू डू द क्लिनिकल वर्क, राइट? सो दिस इज बेसिकली रिहैबिलिटेशन, न्यूट्रिशन, फ्यूलिंग फिजियोथेरेपी सक्सेस इन मस्कुलोस्केलेटल रिकवरी. हम सब जानते हैं मस्कुलोस्केलेटल रिकवरी किस तरह से देना है, कैसे देना है, वो सारा हम पढ़ रहे हैं, लेकिन उसको एड कैसे करना है, हाउ टू हेल्प दैट टू गेट फास्ट. मस्कुलोस्केलेटल रिकवरी हमारे फिजियोथेरेपी ट्रीटमेंट्स के साथ में न्यूट्रिशन ऐड करके हम उसके अंदर किस तरह से उसको और फास्ट कर सकते हैं, उसको किस तरह से ज़्यादा जल्दी कर सकते हैं, जिससे पेशेंट रिकवरी हमारे पास में जल्दी होगी तो पेशेंट सेटिस्फेक्शन ज़्यादा होगा, राइट? एक छोटी सी चीज़ ही बात करते हैं अपन. जितने भी फिजियोज हैं, जितने भी ह्यूमन बींग्स हैं, जितने भी लोग हैं, हम न्यूट्रिशन को क्यों निगलेक्ट करते हैं या कैसे निगलेक्ट हो जाता है? एक छोटी सी चीज़ है, एग्जाम्पल दूंगा आपको. एप्पल सब खाते हैं, ज़्यादातर लोग खाते हैं, सबको पता है. कहाँ बनता है? वेयर डू वी हैव दिस एप्पल में जो पेड़ हैं ज़्यादातर? हाँ, कोल्ड स्टोरेज कौन बोल रहे हैं? ओके, या राइट. सो वहाँ से जो पेड़ के ऊपर जब when the apple is on the tree, वहाँ से हमने उसको प्लग किया. प्लग करने के टाइम पर जो न्यूट्रिशनल वैल्यू उसके अंदर है, क्या वो न्यूट्रिशनल वैल्यू आप लोग ले पाते हैं when you consume that? Is it possible? कारण, रीजन? एग्जैक्टली, प्रिजर्वेशन तो है, लेकिन जब उस एप्पल को वहां से तोड़ा, उसके बाद में हम कहां तक पहुंचे? उसको पैक किया गया, प्रिजर्वेटिव्स लगाए गए, उसके बाद में ट्रांसपोर्ट किया गया. ट्रांसपोर्ट करने के बाद में मार्केट में आया, मंडी में आया, और हमने वहां से लिया. खा लिया? नहीं, ले जाके फ्रिज में रख दिया. फ्रिज में रखने के बाद फिर 3-4 दिन बाद उसको कंज्यूम किया. तो क्या लगता है आपको कि उस न्यूट्रिशनल वैल्यू में क्या डिफरेंस आया होगा उस एप्पल के अंदर, जो जब वो पेड़ से टूटा था और जब आपने खाया? 50% भी नहीं रहता ना, 50% भी नहीं रहता, राइट? तो हम लोग क्या है, एवरी डे व्हाट वी आर डूइंग इज वी आर गेटिंग न्यूट्रिशनल डेफिसिट इन द बॉडी. हमारे विटामिन्स, मिनरल्स, प्रोटीन लेवल्स सब कम होते जा रहे हैं, एंड दैट्स व्हाई वी आर बिकमिंग अ डिजीज्ड कंट्री. आपको याद है इंडिया में कभी भी कोई बहुत ज्यादा डिजीजेस हुआ नहीं करती थी पहले के समय में. क्यों? क्योंकि बहुत सारी चीजें जो है वो हमारी प्योर होती है, राइट? सॉइल कंटैमिनेटेड, वाटर कंटैमिनेटेड, हाँ, और ये दोनों चीजें जरूरी हैं. हवा, एयर कंटैमिनेटेड, राइट? ऑल दीज 3 थिंग्स आर इंपॉर्टेंट फॉर ग्रोइंग एनीथिंग. बच्चे नीचे देख रहा, सो रहा है बेटा, तेरे फ्यूचर की बात कर रहा हूँ. आगे पैसा कमाना है ना अगर, तो यहीं से आएगा, ठीक है? वी हैव दिस इलनेस वर्टिकल रेजिंग, जहां हम काम कर रहे हैं, गधों की तरह काम कर रहे हैं. और अगर आप फिजियोथेरेपिस्ट हो और आप ये समझते हो कि मेरे पास में बहुत सारी टेक्निक्स हैं, मेरे पास में बहुत सारा ज्ञान है, तो वो ज्ञान दूसरे फिजियोथेरेपिस्ट के पास भी है. 5,000 का वर्कशॉप अटेंड करो, 2,000 का वर्कशॉप अटेंड करो. जो ड्राई नीडलिंग आपको आती है, जो मैनुअल थेरेपी आपको आती है, वो दूसरे को भी आती है. हाउ आर यू डिफरेंट फ्रॉम अदर्स? आज अगर हम लोग बहुत सीनियर हैं, 20 साल हो गए, 15 साल हो गए, और आप जो नई जनरेशन अभी आ रहे हो, हमारे बराबर खड़े हो. कभी बात कर बाद में बात कर लूंगा मैं तो, ठीक है? ओके, सो व्हाट हैपेंस, अभी आज की जो नई जनरेशन है ये आ रही है निकल करके, हमारे बराबर खड़ी हो रही है. ये हमें अब लग रहा है. क्यों ऐसा? मुझे मैनुअल थेरेपी आती है, इसको भी आती है, मुझे ट्रीटमेंट आता है, प्रोटोकॉल आता है, ट्रेडिशनल पता है क्या ट्रेडिशनल ट्रीटमेंट मुझे देना है, ACL का, दूसरी चीजों का, वो इसको भी आता है. हाउ एम आई डिफरेंट फ्रॉम द पीपल हु आर कमिंग नाउ? हमारी प्रैक्टिस कैसे डिफरेंट होगी? हाउ? फिजियोलॉजी पढ़ना शुरू करो, जिसको नेग्लेक्ट करा, पैथोफिजियोलॉजी ऑफ द प्रॉब्लम्स, एंड योर सॉल्यूशन ऑफ मेकिंग मनी लाइज हियर इन फिजियोलॉजी. कुछ समझ पा रहे हैं मैं बात कर पा रहा हूँ? तो ये चीज समझ पा रहे हैं या ये सब भूख लगी है? ओके. अच्छा, एक चीज ये भूख से मेरे को याद आया, हम लोग बहुत बार ये बात करते हैं कि गट फीलिंग, ये क्या है? ये गट फीलिंग क्या है? व्हाट इज दिस गट फीलिंग? क्यों बोलते हैं हम? सोचते कहां से हैं? यहां से कि यहां से? वैसे तो सोचते यहां से हैं, लेकिन गट फीलिंग यहां से क्यों आती है? किसी ने सोचा इस बारे में? Why gut feeling? Anybody has any answer? You have the mics. Make it more interactive. अपन बात कर रहे हैं, ये देखो कॉन्फ्रेंस में आप आते हो, सुनते हो, कितना समझ में आता है वो मुझे भी पता है, क्योंकि मैंने भी अटेंड करी अपने बैचलर्स लाइफ के अंदर ये सारी कॉन्फ्रेंसेस. मास्टर्स टाइम में कॉन्फ्रेंसेस we are attending now also. So how much you go and understand and how much you take back, वो important है, right? तो एक बार इसको थोड़ा सा इंटरेक्टिव करते हैं. What is gut feeling? कोई भी क्यों बोलते हैं गट फीलिंग? अच्छा, सेकंड ब्रेन क्या है हमारी बॉडी का? गट. क्यों? 100 मिलियन न्यूरॉन्स जो हैं वो इस गट के अंदर, इसलिए हम बात करते हैं गट फीलिंग की. यहाँ डाइजेशन ऑटोमेटिक हो रहा है, ऑब्जॉर्प्शन अपने आप हो रहा है, सेंट्रल लाइन से उसका कोई लेना देना नहीं है, सेंट्रल ब्रेन जो है वो उसको कंट्रोल नहीं करता है. दैट्स व्हाई वी कॉल दिस एज सेकंड ब्रेन, राइट? कितने लोग हैं जिनको ये आज पता लगा? सच हाथ खड़ा करो, कोई दिक्कत नहीं, आई एम नॉट जजिंग एनीबॉडी. देखो 1 2 3 4 5 6 7 क्वाइट अ लॉट. ओके. तो ये जो हमारा गट है, अगर ये खराब है, अगर गट और लिवर, जिसे हम बोलते हैं गट लिवर एक्सिस, आई एम सॉरी, आई विल कम इन इंग्लिश आल्सो. ओके. सो व्हाटएवर द गट लिवर एक्सिस इज दैट, अगर दिस इज डैमेज्ड और दिस इज deprived of the protein, the macronutrients or the micronutrients, what will happen? Ma'am has given a very good insight already. The brainstorming session was there earlier. It makes me easy to explain. Okay. So what will happen with the protein deficiency? Exactly, muscle quality will deplete. Okay. What will happen to the liver? Any idea? यही प्रॉब्लम है. हम फिजियोलॉजी सब्जेक्ट को सब्जेक्ट की तरह पढ़ते हैं कि पास करना है हमारे को उसको खाली, उसकी पैथोफिजियोलॉजी, उसकी जो पीछे की बिहाइंड की साइंस है ना, वहाँ रखा है सारा पैसा. लगे पड़े हैं, पैसे मूवमेंट कर रहे हैं, लगे पड़े हैं उसके अंदर, PNF कर रहे हैं, दूसरी चीजें कर रहे हैं. अरे यार, इस इलनेस वर्टिकल के साथ में एक वर्टिकल आजकल खड़ा हो गया पूरा. इंस्टा पे आप लोग देखते हो क्या ये सब चीजें? अपन लोग इंस्टा बहुत देखते हैं, सब चीजें देखते हैं. वी आर देयर इनटू दिस वेलनेस, 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 सब तरफ वेलनेस. Protein supplements are getting, omega-3 supplements are there. Then you have calcium, you have magnesium, you have zinc, yes, zinc, yes, creatine, yes. The gym industry, वहाँ पर supplements में आप जाओगे ना, you have big shops. तो ये supplements only gym going के लिए important है?
No, sir.
It is important for everyone. Why you are getting diabetes? Why you are getting various weaknesses, Parkinson's? They have got various things as stroke, cardiac abnormalities and cardiac problems. ये आजकल इतनी ज़्यादा क्यों हो रही है? पहले तो सुनने में नहीं आती थी. सॉरी, चढ़ते समय बोला था 20 मिनट में कंप्लीट करना है, पहली स्लाइड ही मिली. ओके. So we have these contents which are basic. अपन स्लाइड पे बहुत ज़्यादा फोकस नहीं कर रहे हैं, अपन बात सिर्फ इतनी कर रहे हैं कि यहाँ से हमें ये समझ के जाना है कि प्रोटीन और ये जो मैक्रोन्यूट्रिएंट्स हैं, ये जो माइक्रोन्यूट्रिएंट्स हैं, इनका हमारी बॉडी में क्या रोल है, कितना हम हेल्प कर सकते हैं हमारे पेशेंट की रिकवरी पे. क्योंकि जितना पेशेंट का रिकवरी जल्दी होगा, उतना पेशेंट हमारे पास पब्लिसिटी ज़्यादा होगी. माउथ पब्लिसिटी आप कितनी भी कर दो, अखबार में ऐड दे दो, पैम्पलेट्स दे दो, those publicity doesn't matter. Only matters is माउथ पब्लिसिटी, पेशेंट ने जाके पेशेंट को बोला तो 10 पेशेंट आएंगे, और तुमने खराब किया ना तो एक तो झापड़ मारेगा, दूसरा ये बोलेगा भैया इसके पास मत जाना, 100 पेशेंट खराब करेगा, राइट? सो यू हैव टू, दिस सेशन इज एंटायरली फॉर यू स्टूडेंट्स टू अंडरस्टैंड कि अगर हमने फिजियोलॉजी पढ़ा तो मनी विल फॉलो लाइक एनीथिंग। यहाँ कितने लोग हैं जो बाद में फ्री की सेवा देने वाले हैं फिजियोथेरेपी में? हाथ पर रखा, कोई भी नहीं? ऑल ऑफ यू हियर फॉर मनी, इजंट इट? अरे बाबा, डॉक्टर बनने के बाद फ्री सेवा करने वाले हो क्या? धर्म खाता खोल रहा है? नहीं ना? क्या करोगे? ओपनिंग ऑफ द क्लिनिक फॉर द फाइनेंसेस, राइट? फाइनेंशियल्स चाहिए हमारे को, तो वो फाइनेंशियल्स यहां पड़े हैं, गधा मजदूरी में नहीं है। अंडरस्टैंड? ओके? ओके, सो द कोर कंसेप्ट जो हम बात कर रहे हैं, दैट इज फिजियोथेरेपी प्रोवाइड्स द मैकेनिकल स्टिमुलस एंड द न्यूट्रिशन प्रोवाइड्स द सबस्ट्रेट। अंदर बॉडी में तो उसके पास, बेचारे के पास में है ही नहीं कुछ, कंटेंट ही नहीं है, प्रोटीन डेफिशिएंट है, विटामिन ए डी कैल्शियम सब डेफिशिएंट है, आप लगे पड़े भैया ये कर, दिन में ये कर, दिन में 10 बार ये कर। बॉडी में सबस्ट्रेट होगा तो वो कर पाएगा, वो आपके दूसरे दिन बोलेगा सर आपने जो बताया था हो नहीं पाया, वीकनेस थी बहुत ज़्यादा, आई वॉज नॉट एबल टू डू इट। हाउ डू यू कैटर दैट वीकनेस? Multivitamins, multiminerals are needed. I'm not saying that we are replacing the dietitians. No, I'm nowhere to replace a dietitian or anybody else. But self growth कैसे आएगा? हमारा काम कैसे होगा? हमारा काम ज़्यादा अच्छा कैसे होगा? काम तो सबका चल रहा है। ऐसे फिजियोथेरेपिस्ट भी हैं जो 5,000 कमा रहे हैं, ऐसे हैं जो 50,000 कमा रहे हैं, और ऐसे भी हैं जो 5 लाख कमा रहे हैं। आपको देखना है ये चीज, आप किस कैटेगरी में आते हैं, 5,000 में, 50,000 में, या 5 लाख में? कितने लोग हैं जो 5 लाख की कैटेगरी में जाना चाहते हैं? ये ऐसे क्यों आप नहीं चाहिए क्या 5 लाख? तो ये ऐसे हाथ उठाया करो पूरा। रुपए बुरे लग रहे हैं क्या आते हुए? नहीं, राइट? तो ये बहुत एक इंपॉर्टेंट कंसेप्ट है जिसपे कि आज मैं बात कर रहा हूँ, और ये आप लोगों के लिए बात कर रहा हूँ। हम लोग तो अब चलने वाले में से हैं, जाने वालों में से। आप लोग आ रहे हो, यू आर इन द फील्ड, यू आर क्रिएटिंग द फील्ड नाउ, ठीक है? ओके, तो जो गैप हमारे को मिल रहा है वो क्या मिल रहा है? मस्कुलोस्केलेटल रिहैबिलिटेशन, इट ऑफन फोकसेस ओनली ऑन मैकेनिक्स, दैट इज लोड, एक्सरसाइजेस, वेरियस थिंग्स, राइट? बट नेग्लेक्टिंग द मेटाबॉलिक कॉस्ट ऑफ रिपेयर। तो क्या मेटाबॉलिज्म के अंदर जो रिपेयर का जो पावर है वो न्यूट्रिशन के बिना आ सकता है? इज इट पॉसिबल? नो सर। तो न्यूट्रिशन इज द इंटीग्रल पार्ट जिसका हमारे को ध्यान रखना है, और हम अक्सर उसी को नेग्लेक्ट करते हैं, राइट? तो अपना ऑब्जेक्टिव क्या है मेनली? लिखा हुआ है ना? ऑब्जेक्टिव क्या है? पैसिव रिकवरी या एक्टिव? Active metabolic support is the main objective, is it not? So the metabolic demands of injury and immobilization. जैसे immobilization हुआ, क्या होगा? What will happen next to just immobilization? Whenever there is an injury, immobilization has to be there. आप करोगे, दूसरा कोई करेगा, या पेशेंट खुद करेगा? कहीं दर्द हो रहा है, कुछ हो रहा है, तो हम उसको काम में लेते हैं क्या? नहीं, पकड़ के बैठे रहते हैं। So that is a form of immobilization. You don't need a plaster cast or don't need actually anything. स्लिंग की जरूरत नहीं है, हम हाथ में हाथ रख के बैठे हैं, वो भी एक immobilization है, राइट? तो क्या करेंगे? immobilization होते ही क्या हो जाएगा? कुछ तो बोलो, माइक हो गया? अरे कोई है क्या माइक वाले भैया हमारे? कोई एक स्टूडेंट वॉलंटियर कर लो, टेक द माइक एंड जस्ट whosoever is interested in giving the answer. Trophy?
Atrophy, muscle atrophy.
Muscle atrophy, right. What else? Decreased blood circulation, right. What else?
Range of motion restricted.
Great. Range of motion restricted. और कुछ समझ आ रहा है या ऊपर से जा रहा है? आ रहा है, थैंक यू। तो क्राइसिस क्या है हमारा? वो है एनाबॉलिक रेजिस्टेंस। एनीबडी हु कैन आंसर मी दिस एनाबॉलिक रेजिस्टेंस, व्हाट इज एनाबॉलिक रेजिस्टेंस? एनीबडी whosoever, एनाबॉलिक रेजिस्टेंस क्या है? क्या है? ये बहुत बड़े टर्म्स नहीं है, नाम ऐसा है, समझने में बहुत इजी है। क्या है एनाबॉलिक रेजिस्टेंस? यस
Sir, the inability of the body after the injury to take up the exercise or to take up the proteins or macronutrients which is being provided to the body.
So how that resistance is there?
Sorry?
How is the resistance of it?
So the post-injury, our body denies to take up the load because of the high inflammation.
Exactly. इंजरी हो गया, उसके बाद में न्यूट्रिशन भी नहीं जा रहा और एक्सरसाइजेस भी टेकअप नहीं हो रही, तो हाउ यू गेट द इम्प्रूवमेंट? सो दैट इज कॉल्ड एज एनाबॉलिक रेजिस्टेंस। इसको कम करने के लिए हमें न्यूट्रिशनल इनपुट्स पेशेंट्स को देने पड़ेंगे, तभी आपके जो एक्सरसाइजेस के जो इनपुट्स हैं वो उनको ज़्यादा फायदा करेंगे, दैट विल बी मोर हेल्पफुल। सो बेसिकली दैट इज द थिंग। Coming to the consequence, that is rapid muscle atrophy, as ma'am said, okay, and degradation will occur within 48 hours of immobilization. So 2-week immobilization can result in loss of lean mass equivalent to 10 years of natural aging. Agreed? Yes. So phases of healing and nutritional targets. What is that? Inflammation do not suppress entirely. उसको जो inflammation को है उनको हमारे को suppress नहीं करना है. That is good thing, as said by sir. Inflammation is good or bad, but inflammation को हमें immediately suppress करने की कोशिश नहीं करना है, since it is having a good effect. So what we need to do is target the anti-inflammatory fats and adequate protein. तो अगर हम प्रोटीन सप्लीमेंटेशन पेशेंट को देते हैं तो वहां से रिकवरी एनहांस होना शुरू हो जाती है, एंड इट गिव्स हेल्प टू योर ट्रीटमेंट प्रोसीजर ऑल्सो, ट्रीटमेंट प्रोटोकॉल ऑल्सो। सेकेंड फेज इज प्रोलिफरेशन फेज और द हाई सिंथेसिस रेट्स। यहां पर हमारा जो बॉडी है वो हील होना शुरू हो जाता है, राइट? सो वी नीड एनर्जी लेवल्स टू बी ऑन द टॉप। विटामिन सी एंड जिंक विल हेल्प अस इन दिस। तो इसके लिए एक चीज और भी जरूरी है कि हमें ये पता हो कि जो सप्लीमेंट हम देना चाह रहे हैं पेशेंट को, उसकी बायो अवेलेबिलिटी और बायो अवेलेबिलिटी बॉडी में कितना है। जनरली जो एनिमल सोर्स या हर्बल सोर्स से हमारे को सप्लीमेंट्स मिलते हैं, उनका जो बायो अवेलेबिलिटी है वो दैट इज समवेयर अराउंड 80 to 98% इन द बॉडी। जैसे प्रोटीन की अगर हम बात करते हैं तो प्रोटीन डाइजेस्टिबिलिटी करेक्टेड अमीनो एसिड स्कोर, पिडिकास की हम बात करते हैं, वी से दैट पिडिकास स्कोर इज 1, दैट मींस दैट प्रोटीन इज अप्रॉक्सीमेटली इक्वल टू 98% डाइजेस्टेबल इन द बॉडी। दूसरे प्रोटीन हो सकता है उतना नहीं है। कई बॉक्सेस आप देखेंगे प्रोटीन के जिसके ऊपर पिडिकास का स्कोर होता ही नहीं है। पिडिकास इज प्रोटीन डाइजेस्टिबिलिटी करेक्टेड अमीनो एसिड स्कोर, right? If that is 1, the protein is highly bioavailable in the body. तो ऐसा सप्लीमेंट आप पेशेंट को दे सकते हैं। Coming to the phase 3, that is remodeling or the tissue alignment and strength. So here we are targeting what? We are targeting collagen, is it not? And protein is the precursor for all this. Coming to the macronutrients, which are the drivers for synthesis. Okay, so muscle protein synthesis, that is MPS versus muscle protein breakdown. Then we have leucine, as said by ma'am. And then we have this older adults and injured tissues require higher amounts or higher doses of leucine. Recommendation is 1.6 grams to 2.3 per kilogram body weight per day. We must overcome metabolic resistance and higher protein balls. तो ये जरूरी है कि हम हायर प्रोटीन डोजेज उनको दें इनिशियल फेज के अंदर, जिससे कि रिकवरी जो है वो ज्यादा हो। क्यों? क्योंकि गिलास तो खाली है ना, प्रोटीन तो खाया ही नहीं अभी तक। हमको लगता है पनीर खा रहे हैं, स्प्राउटेड्स खा रहे हैं, सब चीजें खा रहे हैं, लेकिन प्रोटीन उसमें कितना है और हम कितना खा पा रहे हैं? अबाउट full of paneer, a bowl full of sprouts, not more than 10 grams of protein will be going there. दूध पी लिया, और क्या चीजें हैं प्रोटीन के लिए? What else?
Salmon.
कम है, सैलमन में ओमेगा 3 फैटी एसिड्स और वो सब चीजें ज्यादा है, प्रोटीन थोड़ा कम है। और
Chicken breast.
सारे चिकन खाते हैं क्या? वेजिटेरियंस कितने हैं? मोस्ट ऑफ देम या सोया चंक्स। लेकिन चिकन वाला जो आंसर है वो भी सही है, लेकिन उसके साथ में आपके साथ में फैट कितना था? वे प्रोटीन, यस। पनीर, पनीर से क्या स्माइल अच्छी आती है, इंडियन स्माइल आती है, पनीर से उतना प्रोटीन नहीं मिल पाता लेकिन, ठीक है। हमको लगता है पनीर से प्रोटीन मिलता होगा, लेकिन क्या जो पनीर मार्केट में मिल रहा है उसमें प्रोटीन है? कहां से बन रहा है वो? केमिकल से बन रहा है। और दूध कहां से बन रहा है? दूध जो आता है ओरिजिनली वो कहां से आ रहा है? गाय भैंस जो भी है उनको कौन से इंजेक्शन लगाते हैं? ऑक्सीटोसिन। आपको लगता है वो जो दूध है जो वो गाय चारा खा रही है, वो चारे के अंदर कुछ है कंटेंट जो उसके पेट में जा रहा है जिससे वो अच्छा दूध दे पाएगी? और जो दूध वाला है ना बेचारा आपका, वो जैसे ही दूध निकालते हैं ना, और फिर वो लेके भी आएगा आपके घर पे, आपकी मम्मी या आपके पापा आपको कहेंगे भी दूध पीना है, लेकिन हम पीते क्या? तो जो मिल रहा है ना वो भी नहीं मिल रहा। सो वी आर प्रोटीन डेफिशिएंट डे बाय डे, राइट? ये हमारे जो सफेद बाल हो जाते हैं, वीकनेस आ जाती है, सीढ़ियां चढ़ने उतरने में दिक्कत आती है, नीचे बैठ जाए तो उठने में दिक्कत, इसलिए टॉयलेट हमने हमारे कंफर्टेबल के हिसाब से कर लिया आजकल। इज इट नॉट? तो हमने मॉडिफाई कर दिया अपना एनवायरनमेंट बिकॉज वी वर नॉट एबल टू डू इट। हमने ये नहीं देखा कि वी वर नॉट एबल टू डू इट, उसका रीजन क्या है? व्हाट इज द रीजन बिहाइंड वी आर नॉट एबल टू डू इट? पहले के लोग कर नहीं लेते क्या? वो क्या एक्स्ट्रा कहां से मिल रहा था उनको? इज इट नॉट? तो वो सारी चीजें हमने सोचना बंद कर दिया, हम अपने कंफर्ट जोन में चले गए पूरा। एवरीवेयर वी आर इनटू द कंफर्ट जोन। ओके, so I was talking about protein sources, that is animal source and plant source, as said by you, the whey protein, and then physiology behind is essential amino acids. जैसे मैंने कहा प्रोटीन डाइजेस्टिबिलिटी करेक्टेड अमीनो एसिड्स पेडिकास, ओके। तो उसके अंदर हमारे को करेक्टेड अमीनो एसिड स्कोर मिलता है, विच इज इंपॉर्टेंट फॉर योर मसल प्रोटीन सिंथेसिस, राइट? ओके, गो टू द नेक्स्ट स्लाइड। कार्बोहाइड्रेट्स, इनका क्या रोल है? What is the role of carbohydrates? Yes, energy. Okay. So prevent gluconeogenesis, burning amino acids for the fuel. Sparing the protein, carbohydrates are important. ठीक है? Then glycogen, essential for fueling the rehab session itself. Low glycogen क्या होगा? Low glycogen से क्या होगा?
Blackout.
अभी क्या हो रहा है तुम लोगों के साथ में?
Low energy level.
हैं? भूख लग रही है। Low glycogen levels, energy levels will drop down. Yes. So poor neuromotor output. सर जाओ ना, ये सब हमें आता है। सच्ची बताना, ऐसी फीलिंग आ रही हो तो मैं चला जाऊँ, और अगर नहीं आता है तो सुन लो, आज का ये सेशन तुम्हारे को कमा के देके जाएगा। सच्ची बताओ, अगर मैं रोक दूंगा यहीं पे, आई विल स्टॉप इट हियर। धन्यवाद, थैंक यू। एक ही इनपुट बहुत होता है। ओके, अब इसको गालियां पड़ेगी, ठीक बोला? ओके, कॉम्प्लेक्स वर्सेस सिंपल, दैट इज लाइक वी हैव टू फोकस ऑन व्हाट? वी हैव टू फोकस ऑन लो ग्लाइसेमिक इंडेक्स। लो ग्लाइसेमिक इंडेक्स होगा तो क्या होगा? टू मैनेज इंसुलिन सेंसिटिविटी, राइट? एंड क्या होगा वो एक्टिविटी के अंदर? दैट विल बी लर्निंग। नेक्स्ट, एनर्जी बैलेंस, द कॉस्ट ऑफ अंडर ईटिंग। अब हमारा रोल आता है बहुत सारे लोगों का जो हम अंडर ईटिंग करते हैं पतला होने के लिए, अच्छा दिखने के लिए, कपड़ों की फिटिंग हमें अच्छी आए, हैं? सो वी डू व्हाट? अंडर ईटिंग, इज इट नॉट? और क्या होगा उस अंडर ईटिंग का इफेक्ट? हम ऑलरेडी डेफिशिएंट हैं, वी आर ऑलरेडी डेफिशिएंट, एंड नाउ वी आर बिकमिंग more deficient because of that under eating. शादी होने वाली, दाग सर्कल्स खत्म ही नहीं हो रहे यार, क्या करूं? 3 दिन में खत्म हो जाएंगे क्या? नहीं होंगे। अगेन वाटर इनटेक नहीं है, 5 लीटर पानी पीना है, 6 लीटर पानी पीना है, लेकिन नहीं, ठीक है भैया हम तो थम्स अप पिएंगे, हाँ, वी हैव कोल्ड ड्रिंक इंस्टेड ऑफ वाटर. इज इट गुड? इट इंक्रीजेस योर ग्लाइसेमिक इंडेक्स. वाटर नहीं पीना है, हमें दूसरी चीजें नहीं करनी है, एक्सरसाइज प्रॉपर नहीं करनी है और अंडर ईटिंग करनी है. पतला होना है, शादी में अच्छा होना चाहिए, लहंगा जो है ना मेरा वो फिट होना चाहिए, है ना. इसको यूज कम करना, पतली हो जाऊंगी 1 महीने में, तो इसको छोटा कर, इसको टाइट कर थोड़ा और, मैं पतली हो जाऊंगी, मुझे पता है, मैं खाऊंगी नहीं, 2 दिन खाना नहीं खाऊंगी पतली. लड़कों का भी सेम है, तुम भी ज्यादा वो मत करो, है ना. शेरवानी को यहाँ से मैं अपने पेट अंदर करना, तुमको सिला अच्छा दिखना चाहिए. So that type of mentality we are developing on the cost of our body. हमारी बॉडी जो है वो ऑलरेडी वीकेंड है, we are having all those problems, but we are making it more weakened just by doing all these for the cosmetic appearance. Right? मत करो प्लीज, आगे दिक्कत आएगी. एनर्जी बैलेंस के अंदर हम बात कर रहे थे रिलेटिव एनर्जी डेफिशिएंसी इन स्पोर्ट्स. So that is applicable to the rehab patients also, is it not? Then the risk, caloric deficit impairs collagen synthesis and bone turnover. Then we have guideline maintenance, that is calories plus injury factor. Do not diet. Please, I request, do not make the patient, or if you come to know that patient is dieting, do not ask them not to diet during this active recovery phase, acute recovery phase, right? Okay, coming on to the fats. As said by my friend, omega की हम बात कर रहे थे सर आपसे, right? So we have come to this fats, inflammation and inflammation modulation, wherein omega 3, that is EPA and DHA. जब बच्चा छोटा होता है ना, तो उसको EPA और DHA के हम लोग सप्लीमेंट्स देने की कोशिश करते हैं, जिससे कि उसका ब्रेन का जो रिकवरी है वो ज़्यादा हो. इसे बात करते हैं ना, EPA DHA सुना है? हाँ, yes. So potent and anti-inflammatory properties via resolvent and protecting production. The mechanism is down regulates pro-inflammatory cytokinins IL-6, as said by Mann. Anabolic sensitivity. Evidence suggests omega-3s may sensitize muscle to amino acids. So again, omega-3 are important for protein absorption also and combining anabolic resistance. इसको स्किप कर सकते हैं, टाइम थोड़ा ज़्यादा हो रहा है. दिख रहा है, भूखी नजरों से देख रहे हो मेरे को, बाहर नहीं करके देखता हूँ, खाने के बीच में मेरे बीच में खड़ा था. The tissue, collagen, signs of health. Okay, so now we have this tissue, that is tendons and ligaments are ECM dominant and not cellular dominant. What is ECM? Extracellular. Students के लिए session है. ECM is extracellular matrix, और ये क्या है? Cellular dominant. So this is ECM dominant and not cellular dominant. Mechanism, hydrolyzed collagen peptides plus vitamin C अगर हम देते हैं, तो maximize collagen synthesis हमारा होगा उसके अंदर. दूसरा है प्रोटोकॉल के अंदर हम जाएं तो 15 ग्राम 15 ग्राम ऑफ जिलेटिन, भाई मुझे भी दे देना है. Thank you. Okay, so 15 grams gelatin collagen plus 50 milligrams vitamin C taken 45 to 60 minutes before loading the tendon. Peak blood levels must coincide with mechanical loading to drive peptides into the avascular tendon tissue. कितनों को समझ में आया? देखो, अगर ऐसे शांत बैठोगे ना तो उसका एक मतलब होता है, या तो सब समझ में आ गया और या फिर उड़ गया. मैम हम लेक्चरर्स के साथ में बड़ी प्रॉब्लम है ये बच्चों की, क्लास में नहीं जाते हैं ना, तो बेचारे बैठे रहते हैं, बाद में बोलते हैं यार पता नहीं क्या है, यार पता नहीं क्या पढ़ा देंगे, एग्जाम में क्या लिखेंगे. अरे तो पूछ लो ना यार उसी समय, बाद में सोचते हो. या एंड द टीचर इज थिंकिंग हाँ सब समझ में आया. मेरी क्लास में सारे शांत बैठे थे, हम लोग फूल के एकदम ऐसे बाहर निकलते हैं क्लास से. आज का सेशन मैंने बहुत अच्छा लिया, आज की क्लास मैंने बहुत अच्छी ली, बच्चों को सब समझ में आया. वहाँ पता ही नहीं पीछे जाना कर रही हमारी. एक चीज आपको सजेस्ट करूंगा, बहुत सारी चीज़ें होती हैं जो हम अगर स्टोरी के रूप में पढ़ें तो वो हमेशा ज़्यादा याद रहती है, ठीक है। और अगर हम बोलें उसको, डार्विन की थ्योरी किस किस को याद है? अच्छा चलो ओम्स लॉ बता दो बच्चे। है ना? और ये अगर हम इसको थोड़ा सा स्टोरी के रूप में पढ़ लें ना तो थोड़ा सा याद रहेगा, है? तो स्टोरी बनाना भी इम्पोर्टेंट है पढ़ाई के अंदर। चलो, माइक्रोन्यूट्रिएंट्स, इनका रोल कुछ आइडिया है किसी को? विटामिन ए क्या करता है, चलो बताओ कोई भी। कहां पर, कहां पर? यहाँ पर नहीं, कहां पर? रेटिनल प्रोटेक्शन कहां? ओके, विटामिन डी क्या करता है? क्या ए ऐसे नहीं, एक माइक दो मैम को दो। मैम विटामिन डी बता पाएंगे ना क्या होता है, क्यों काम में जरूरी है हमारे बॉडी में? दो ना माइक दो मैडम। सिट सिट सिट, जबरदस्ती तो नहीं पूछे, इंपॉर्टेंट है। क्या करता क्या है विटामिन डी? बॉडी में पड़ा हुआ है विटामिन डी, करेगा क्या? वैसे तो है ही, क्योंकि अंदर ही बैठे रहते हैं हम तो सारे, एसी छूटता ही नहीं हमारे से। गाड़ी में एसी, रूम में एसी, घर में एसी, बाहर निकलते गए। हाँ, विटामिन डी इज अ प्रिकर्सर फॉर द रिएक्शन टू हैपन वेयरइन सीरम कैल्शियम, ब्लड में जो कैल्शियम पड़ा हुआ रहता है ना, हम टेस्ट करवाते हैं, सीरम कैल्शियम का टेस्ट करवाते हैं, तो वो जो बोन में ट्रांसफर करने का काम है, इट एक्ट एज अ कैटलिस्ट, कोलेकैल्सिफेरॉल, राइट? ये बात आज किस किस को पता लगी पहली बार? मतलब मेरे यार। ओके ओके, तो ये सब चीजें इंपॉर्टेंट है पढ़ना। और हम क्या करते हैं, फिजियोथेरेपी को बहुत लाइटली ले लें, जबकि आप वो लोग हो जो कि न्यूरोसर्जन्स, न्यूरोफिजिशियंस, ऑर्थोपेडिशियंस और सबके सामने ना यूं नजर से नजर मिला के बात कर सकते हो। लेकिन करते क्या हो, जब वो बोल रहा होता है तो हाँ सर, ओके सर, यस सर, हो जाएगा सर, उन्होंने जो बताया कर लिया। बेटा वो स्टेट नहीं लगा, अब एनसीएचपी आ गया, तो अपना स्टेट बहुत अलग होने वाला है, हो भी गया और होने भी वाला है। तो उस तरह का लेवल ऑफ नॉलेज आपको अपने पास रखना पड़ेगा कि आप उनसे आंखों में आंखें डाल के अपना कंसेप्ट जो है उनको बता सको। तो हो सकता है उनको भी ना पता हो, क्योंकि वो भी आपकी जैसी हैं, अभी निकले। कई चीजें जैसे सर ने बोला, सर लोग बताते हैं, फार्मासिस्ट पीपल दे से द व्हाट मेडिसिन दे हैव टू राइट एंड दे राइट दैट मेडिसिन। उनको भी नहीं पता है, ये मुझे मेरी क्लिनिकल प्रैक्टिस में पता लगा 2006 में, when I started with the clinical practice, so I came to know that even the orthopedicians and neurosurgeons go along with the pharmacy people who just come to their clinic, explain the drugs कि इसका ये रोल है, इसका ये रोल है, and write this drug। तब उनके समझ में आती है पीके के लिखते हैं, ये फायदा भी है, फॉरेन ट्रिप भी मिलेगा। सही कह रहे उनको भी नहीं पता है। तो अगर आपने पढ़ लिया ना, वाह नहीं कर सकते। At my clinic, orthopedicians and neurosurgeons used to come take my treatment. 2006, 3 physiotherapists थे कोटा में कल। I belong to Kota, Rajasthan. So only 3 physiotherapists थे वहाँ पे, and we created the market of physiotherapy over there. वो कहानी सुनी है? एक कंपनी थी चप्पल वाली, उसने एक आदमी को भेजा एक गांव के अंदर और उसने कहा कि देख के आओ अपनी चप्पल कितनी बिकेगी। तो उसने क्या कहा, ठीक है सर जाता हूँ। गया, वापस आया, सर एक ही चप्पल नहीं बिकेगी वहां, तो लोग जानते ही नहीं चप्पल क्या होती है। उसने एक दूसरे आदमी को भेजा, दूसरा आदमी गया, बोले सर चप्पल बहुत बिकेगी, क्योंकि वहां लोग जानते नहीं है चप्पल क्या होती है, तो सबको पहनाएंगे सर। अंडरस्टैंड द कंसेप्ट। फिजियोथेरेपी अवेयरनेस अभी बहुत आ चुकी है, आप हमारे टाइम में नहीं हो जहाँ पे हम तो अवेयरनेस क्रिएट करने में लगे हुए थे। अभी इतनी अवेयरनेस आ गई जिसकी हद नहीं, पेशेंट खुद ही आता है। लेकिन हाँ एक प्रॉब्लम है पेशेंट के साथ, गूगल करके आता है ना। आके बोलता है बेचारा ये कर दो ये कर दो ये कर दो। एक ही ब्रांच है ना वो अभी तक बची हुई है इससे, कौन सी? पेशेंट कांट गूगल वेटनरी, है ना? वेटनरी में पेशेंट कांट गूगल, सो दैट इज द ओनली ब्रांच लेफ्ट। अदरवाइज तो पेशेंट गूगल करके आएगा, मुझे ये है ये है ये क्या ये करवा दो आपसे। चलो नेक्स्ट, हाइड्रेशन की बात हम कर चुके हैं, वाटर इनटेक इज वेरी इंपॉर्टेंट, ठीक है? आ गया कंक्लूजन, खत्म। ओके, ये पार्ट थोड़ा समझ लो अच्छे से। ओके, वी हैव न्यूट्रिशन प्रिस्क्रिप्शन। अरे इंटरेस्टिंग है सेशन या समझ आ रहा है? कुछ भी थोड़ा बहुत मैं कह रहा हूँ, एक ही पॉइंट दिमाग में लेके जाओगे ना तो उसी को आगे बनाओगे तो विल मेक मनी। सो प्री रिहैब, दैट इज कॉम्प्लेक्स कार्ब एंड फ्लूइड, देन वी हैव पेरी रिहैब, दैट इज ड्यूरिंग द रिहैब पार्ट। So if the session is 60 minutes, water and electrolytes are important. Then post we have leucine rich protein, carb for the repair. Daily base, that is anti-inflammatory fats, sufficient micronutrients, total energy balance. Interdisciplinary collaboration, that is the team, that is always we say it's a teamwork. Then we have physician, physiotherapist and dietitian. शुरुआत में मैंने कहा we are not replacing the role of डाइटिशियन, पर अगर हमारे अंदर नॉलेज है न्यूट्रिशन की तो बुरी नहीं है, अच्छी है, कहीं ना कहीं काम आएगी। टेक होम मैसेज क्या है आपका? ये मैकेनिकल लोडिंग इन द स्पार्क न्यूट्रिशन इज द फ्यूल, यू कैन नॉट ट्रेन अ पुअर मेटाबॉलिक एनवायरनमेंट। बेस ही नहीं है तो आपकी ट्रेनिंग कहाँ से होगी? मतलब नींव ही नहीं डली तो बिल्डिंग कैसे बनी? राइट, कॉल टू द एक्शन, दैट इज इंटीग्रेट basic nutritional screening into your initial musculoskeletal assessment. So I rest myself. आपको कोई सवाल पूछना है तो अलग से पूछ सकते हैं, जिनको लंच के लिए जाना है लंच के लिए जा सकते हैं। थैंक यू।
Any questions from the audience?
Okay. Yeah. Sir. Pardon me, sir? How am I looking today?
Very good.
Right? So I was much better because I am taking supplements since 2006. I am 47. कैसे लगना चाहिए ना, मैं बता रहा हूँ ना, मैं अभी आज कैसा हूँ शादी में। much better, I was much better looking in the wedding, for my wedding, right? And now I am 47 and I am taking the supplement since 2006.
My question is, how do you stop the patient from taking too many supplements?
That guided dosage has to be followed with the knowledge of prescription, with the knowledge by study, and with the knowledge that the dosage given onto the pouch. That is important because different supplements have different contents in per milligram or mcg, microgram. So you have to follow that dosage judiciously.
You also said that patients Google a lot.
Yeah.
And what we find in Australia is that patients overdose on supplements, poisons.
Right.
So I think your role is that part of
That is again a multi-vast topic because poisoning from the supplement, that is again a very good suggestion from sir. We have to take care of that also क्योंकि कई बार होता है लोग सोचते हैं मैं जल्दी जो है वो recover कर जाऊँगा, I'll be taking more supplements that will recover me more faster. So that is not the truth, that is a myth, and we have to judiciously use the supplements. So again you are the guiding factor for that or the dietitian. Okay. Right. Thank you. Thank you. Thank you. Yeah?
Congratulations, sir. Fantastic talk.
Thank you, sir.
I just want to interact one thing to the students here. Sir की talk से there are two things which we as a clinician should take home. Number one, recovery phase is more important than burnout. What all exercise we are prescribing to our patients, therapeutic interventions, we are giving it for the purpose of reversing the functional SCR, which is our main treatment goal. Now, as a movement scientist, we need to understand that every single cellular mechanism requires fuel, and we are what we eat. So, what all things we are eating will define the outcome of the movement. This is modern perspective. Coming to the old ancient traditional Indian medicine, which is Ayurveda, often in Ayurveda it is being quoted as the definition of health from point of view of nutrition is Pair garam. I am repeating. Pair garam, pet naram, sar thanda. The feet should always be warm. They should never be cold and clumsy. The abdomen means the digestive process should always be supple. There should be no irritation on point of view of hyperacidity or constipation nature. And the mind, the temperament should always be the key. That is the definition of health from point of view of nutrition. They call it 3 doshas. Vata, Pitta and Kapha, the balance. So if there is any imbalance nature which is being caused at the cellular level, as a physiotherapist, it will definitely hamper our outcome. Now it is not included in our curriculum directly. And we have some legal restrictions and boundaries also, ethical purview also. We cannot go and prescribe that. So, we are not supposed to do that also. But yes, we can counsel them or we can collaborate with a better person available. True. Right? And AC Guyton, textbook of medical physiology, states that whatever intervention we do, if the state of the cell is devoid of oxygen, we will never be able to achieve our target. So it is the oxygen which should reach to the cellular level. That goes for that. Thank you so much.
थैंक यू सर, थैंक यू।
और स्टूडेंट्स एक मैसेज और जब सर ने आपसे पूछा 5 लाख रुपए कौन कमाना चाहता है, इतने आलसी हो आप लोग कि पहले तो इतना सा हाथ उठा रहे थे, फिर सर ने जब बोला तो थोड़ा सा हाथ भी उठाया। मैं आपकी जगह होता तो मैं अपने दोनों हाथ उठाता, ईश्वर ने 2 हाथ दिए।
एटलीस्ट मैनिफेस्ट तो करो, कमा नहीं पा रहे हो कोई बात नहीं, मैनिफेस्ट तो करो यार कम से कम कि हाँ हमको 5 लाख रुपए कमाने हैं। अब वो एक दिन के कमाने के महीने के कमाने वो डिसाइड कर लेना।
And on a joke note, lighter note, carry your message is apple कब खाना चाहिए? पेड़ से तोड़ते ही।
उगाओ पेड़ उगाओ। चलो, थैंक यू सर, थैंक यू सो मच, थैंक यू।
Emphasizing overlooking extreme vital and role of nutrition and rehabilitation also. My personal experience is if we got the sun in the morning, we would have slept till long. So thank you so much sir for such a session. I would request the dignitaries and session chair to take a token of appreciation to our esteemed speaker, Professor Dr. Bhupesh Kaur. Is Professor Dr. Associate Professor, Department of Physiotherapy, Swami Rama Himalaya University, London. The topic for the expert is effectiveness of task-oriented and rehabilitation in individuals with spinal cord injury. I request all the students in the auditorium to please be seated. We have very less time. We know that you are hungry. Please be seated.
brainstorming session of self-build yourself. अभी पलक सर ने भी कहा ईश्वर ने आपको 2 हाथ दिए हैं और मैं उससे बात करूंगा अगर हमारे 2 हाथ नहीं है क्या होगा. I have attended so many conferences, national, international. People talk about pain, myofascial pain, backache, brain, neuroplasticity, pelvic floor. But in so many conferences, head के ऊपर कोई बात ही नहीं होती, चर्चा ही नहीं होती. And if you think about our brain is a CPU, our thorax is a software. हमारे पैर that is an emulator. हाथ क्या है हमारे? What is our hand? हम हाथ से क्या कर सकते हैं? We can do innovations from our hand. आप सोचो उस इंसान के बारे में जिसके हाथ नहीं है, पैर है हाथ नहीं है, वो काम ही नहीं कर सकता. तो there is a thing called अगर आपके हाथ नहीं तो काम नहीं, और अगर आपके हाथ है तो साथ है. Same way I will like to talk about today's presentation. When spinal cord injury occurs, 2 चीजें होती हैं, या तो टेट्राप्लेजिया या पैराप्लेजिया. आई थिंक उसको पता होगा, या तो आपके दोनों पैर चले जाते हैं या आपके दोनों हाथ दोनों पैर चले गए. अब हमारा मेन फंक्शन क्या होता है हैंड का? व्हाट इज द मेन फंक्शन ऑफ हैंड? एनी स्टूडेंट कैन आंसर. मेन फंक्शन कोई बता सकता है मुझे? लोगों को पता ही नहीं होता हाथ का काम क्या होता है. anyone can tell what is the main function of hand? No, not reach out. Anyone? कोई कहेगा मैनिपुलेशन, कोई कहेगा सर कॉम्बिंग, कोई कहेगा ईटिंग. मेन फंक्शन क्या है? स्टिल अभी भी जो आप लोग कर रहे हो राइट नाउ, मेन फंक्शन है सेंसेशन. आप वो चीज फील कर सकते हो. अगर आपके हाथ ही नहीं है, आपको पता ही नहीं आपकी चाय गर्म है या ठंडी है, आपका ये वुड है या टेक्सचर सॉफ्ट है या हार्ड है. तो मेन फंक्शन ऑफ हैंड इज योर सेंसेशन. एंड इफ आई टॉक अबाउट स्पाइनल कॉर्ड इंजरी, जो डाटाबेस है अभी रिसेंटली 2 साल पहले का 2022 का 23 का, आप देखोगे यूएस में जो हैंड एससीआई की जो इंजरी हुई है वो 22,000, और उसमें जो हैंड पे जो काम हुआ है दैट इज मिलियंस. बहुत वही चर्चा हम इंडिया में कर रहे हैं. इंडिया में हैंड पे काम तो हुआ है मगर उसकी कोई चर्चा नहीं करता. दूसरी चीज, जो हमारा हैंड जो इंजर होता है कब होगा? जब आपको क्वाड्रिप्लेजिया हो गया, टेट्राप्लेजिया हो गया. वो कब होगा? जब आपको सर्वाइकल कॉर्ड इंजरी, हाई सर्वाइकल कॉर्ड इंजरी C1 to C4 लेवल. अगर जो स्टूडेंट को न्यूरो में इंटरेस्ट है उनको पता होगा C1 to C4 इज द हाई सर्वाइकल कॉर्ड इंजरी, और उसमें C5 लेवल इज द मोस्ट कॉमन. तो वहां पे आपका हैंड रिहेबिलिटेशन का काम बहुत ज्यादा होता है. सेकेंड थिंग, अगर आपका हैंड इंजर हो गया, आई हैव टोल्ड ऑलरेडी, आपके सारी एजियर एक्टिविटीज, आपका एक्सप्रेशन, इस टाइम मैं बोल रहा हूं, मैं अपना हाथ हिला रहा हूं, मैं क्या कर रहा हूं? आपको एक्सप्रेस कर रहा हूं. ठीक है? आपके ईटिंग, कुकिंग, कॉमिंग सब चला गया. सो व्हाई हैंड रिहेबिलिटेशन इंजर नेसेसरी? क्वेश्चन ये है, आई टेल यू वन थिंग, व्हेन आई यूज्ड टू स्टडी इन माय बीपीटी, बैचलर्स मैंने बैंगलोर से किया, मेरे एक टीचर ये कहते थे, फॉर अ फिजियोथेरेपिस्ट देयर आर ओनली 2 एसेट्स. केवल एक फिजियोथेरेपी का 2 चीजें होनी चाहिए, एक आपकी कमांड, एक आपका हार्ट. आपका अगर मैनिपुलेशन अच्छा है, आपका मोबिलाइजेशन अच्छा है, यू कैन चेंज द वर्ल्ड, यू कैन डू इनोवेशंस, यू कैन डू रिसर्च, और आपकी कमांड अच्छी है तो. और हैंड कहाँ काम आता है आपको देखो हर प्रोफेशन में, वेदर यू आर टीचर, वेदर यू आर मल्टीनेशनल वर्कर, वेदर यू आर सॉफ्टवेयर इंजीनियर, वेदर यू आर कारपेंटर, एंड वेदर यू आर इवन अ स्किल वर्कर, हर जगह हैंड रिहेबिलिटेशन काम आता है. आप सोचो उस इंसान के बारे में जिसको टेट्राप्लेजिया हो गया, चारों हाथ, उसका घर कैसा चलता होगा. तो मैं दैट्स व्हाई आई हैव चूजन दिस टॉपिक. हैंड रिहेबिलिटेशन इज अ वेरी यूजफुल टूल एंड यू कैन अर्न योर अर्निंग. आप सोचो जिसका घर नहीं चलता कैसे काम करेगा वो? एक लिफ्टमैन है, एक कोई बाहर सिक्योरिटी गार्ड है. आप सोचो अगर उसको उसके दोनों हाथ नहीं है, पैर है मगर हाथ नहीं है, क्या वो काम करेगा? So it is a very you have to think beyond that. That's why I have chosen this topic. Next, now thing is that हमारी जो स्पाइनल कॉर्ड इंजरी होती है दो टाइप की होती है, एक कंप्लीट एक इनकम्प्लीट। कंप्लीट इंजरी में तो कोई प्रोग्नोसिस होती नहीं है, दैट इज एशिया ए ग्रेड, पता होगा आई थिंक। इनकम्प्लीट इंजरी में कुछ थोड़ा बहुत एक्सपेक्टेशन कर सकते हैं, जो अब ट्रांसफॉर्मेशन में सेंसरी मैपिंग, कॉर्टिकल रिऑर्गेनाइजेशन, न्यूरोप्लास्टिसिटी और देयर सो मेनी वर्क हैज बीन डन, बट हम लोग बात ही नहीं करते उस पर। तो जो स्पाइनल कॉर्ड इंजरी की जो गाइडलाइंस है वो ये कहती है कि जो हमारी डिफॉर्मिटीज होती है हैंड में, कई डिफॉर्मिटीज होती है, पेशेंट इवन कई बार सीटीएस में भी चला जाता है, पेशेंट को सीटीएस है नहीं, पेशेंट को सीटीएस हो गया, पेशेंट को अपना कोई डिफॉर्मिटीज हो गई, कॉन्टेक्चर्स हो गए, तो उसका रिहैबिलिटेशन कैसे करें, दैट इज द मेन थिंग ऑफ फोकस। नेक्स्ट थिंग, अब हैंड रिहैबिलिटेशन आपको करना तो है, मगर उसका एक्सेसिबल करना नहीं आता। मेनी पीपल दे डोंट नो हाउ टू एसेस दैट थिंग। तो इन स्पाइनल कॉर्ड इंजरी we usually focus on two grips, एक होती है palmar grip, एक होती है pinch grip, and that is very important one। pinch grip सबको पता होगा, pinch grip is something like that, ठीक है, आप कोई चीज उठा रहे हो, पेन उठा रहे हो, ऑलपेन उठा रहे हो, समथिंग, palmar grip is like this, okay। और स्पाइनल कॉर्ड में ये दो ग्रिप बहुत काम आती है। आई विल टेल यू इन अभी आगे आपको बताऊंगा हाउ दिस वर्क। Now, if you talk about this, these are the ये स्केल है सोलरमैन हैंड फंक्शन टेस्ट। ये एक असेसमेंट टूल है स्पाइनल कॉर्ड इंजरी के बारे में। ये कैसे, मैं आपको बता देता हूँ ब्रीफ में, हाउ व्हाट डज दिस टूल कंटेन। इट कंसिस्ट ऑफ सम ग्रिप्स, मीनिंग दिस ग्रिप, पिंच ग्रिप, इंडेक्स ग्रिप, पाल्मर ग्रिप, ऑल दिस ग्रिप, और इसमें 20 टेस्ट होते हैं, 20 सब टेस्ट। दीज आर रिटन हियर, हाउ टू वॉश द क्लोथ्स, हाउ टू ईट, हाउ टू थिंक, इसका इवैल्यूएशन करना पड़ता है, एंड दैट इज फ्रॉम 0 to 4 स्केल। अगर पेशेंट ने सब कुछ किया दैट इज 0, अदरवाइज इट विल बी 4। तो दैट इज अ इंपॉर्टेंट टूल हाउ इट वर्क्स। अनदर टूल है दैट इज CUE, कैपेबिलिटीज ऑफ अपर एक्सट्रीमिटी क्वेश्चनर। सो दैट इज अनदर टूल दैट कैन बी स्कोर्ड फ्रॉम 0 to 4, with a 4 is a low difficulty level and 0 patient कर नहीं पा। That is very important tool. That is also done in catheterized patient. These are the some of the questions जो patient से पूछते हैं। We categorize it to mild, moderate, severe, very severe accordingly. Now, thing is that आप उसमें क्या-क्या चीजें कर सकते हो। पेशेंट को कॉन्टेक्चर्स हो गए, स्पार्सिसिटी हो जाती है, सब पे काम करते हैं, कोई स्ट्रेचिंग कर रहा है, कोई मॉडलिटी दे रहा है, हॉट पैक लगा रहा है, बट एक्चुअली कोई सेंसेशन पे काम करता है, नोबडी वांट्स। बहुत रेयर सेंसरी मैपिंग, सेंसरी मैपिंग का डिस्टर्ब हो गया, आपकी न्यूरोप्लास्टिसिटी, आपका कॉर्टिकल रिऑर्गेनाइजेशन हो ही नहीं रहा, तो वहां पे हमें काम करने की जरूरत है। दैट इज वेरी इंपॉर्टेंट। नाउ देयर हैव बीन सम स्टडीज हैव बीन डन which aim to increase the recruitment of muscles above and below the affected level। जहां पे आपका इंजरी हुआ जो लेवल है, हमें उसके ऊपर और नीचे काम करने की जरूरत है। हम क्या करते हैं, जहां पे इंजरी हुई है उसी लेवल पे काम कर रहे हैं। दैट्स व्हाई आवर प्रोग्नोसिस इज नॉट, पेशेंट का प्रोग्नोसिस हो नहीं पाता। दैट इज द थिंग। सो दैट इज अंडरस्टूड। अगर आपका एशिया लेवल ए है, इफ द एशिया लेवल इज ए, पेशेंट प्रोग्नोसिस अच्छा नहीं होगा, बट एटलीस्ट वी कैन वर्क, वी कैन मेक द पेशेंट दैट मच कि पेशेंट का लाइवलीहुड कैन अर्न। दैट इज वेरी इंपॉर्टेंट। सो there have been studies have been done where the work has been done on sensory mapping through neuroplasticity and cortical reorganization, and there have been improvement has been seen in the hand rehabilitation। task specific activities, patient को वो काम कराओ जो उसकी जरूरत है, task specific। अगर if the patient working as a security guard, work for that जो patient को उस टाइम की जरूरत है। if the patient working as a teacher, only specific on the task specific। हम लोग काम करेंगे अदर देन दैट जो पेशेंट का ऑक्यूपेशन वो छोड़ के, तो देन देयर इज नो नीड। पेशेंट का इंप्रूवमेंट हो जाएगा मगर पेशेंट के उस एरिया में इंप्रूवमेंट नहीं होगा। सो हैंड रिहैबिलिटेशन ऑलवेज आल्सो फोकस ऑन हाउ टू अर्न द लाइवलीहुड ऑफ पेशेंट, हाउ टू अर्न द फैमिली, दैट इज द मेन फोकस। FES, nowadays functional electrical stimulation, has been in demand in the physiotherapy। In both these 2 days it has been, इसके बारे में भी बात हुई है काफी। नेक्स्ट थिंग इज प्रैक्टिस कंबाइंड विद स्पॉन्टेनियस मोटर स्टिमुलेशन। प्रैक्टिस जितना ज़्यादा प्रैक्टिस करवाओगे, न्यूरोप्लास्टिसिटी, कॉर्टिकल इम्प्रोवाइजेशन, देयर विल बी सेंसरी रीऑर्गेनाइजेशन, उस पर वर्क होगा। तो पेशेंट को जितनी ज़्यादा प्रैक्टिस कराओगे, either modulating, summarize, patient will work like anything. Electrical instrumentation, yes, electrical instrumentation पे भी कल हम बात कर रहे थे। Electrical instrumentation focuses on the cortical mapping of the head, and it is a new frontier in the spinal cord injuries. Really, it is a much working. Now, this is the important thing. तीनों डिसीज के किसी को नहीं पता होगा। कहीं भी मैं स्पाइनल कॉर्ड इंजरी के बारे में बात करता हूँ, स्टूडेंट्स को पता ही नहीं था व्हाट इज तीनों डिसीज के। दे डोंट नो। सब लोग ये कहते हैं सर उसमें ग्रिप नहीं बन पाती। अरे ग्रिप कौन सी ग्रिप? देयर सर्टेन टाइप ऑफ ग्रिप। तो स्पाइनल कॉर्ड इंजरी में स्पेशली टेट्राप्लेजिया में तीनों डिसीज ग्रिप होती क्या है? तीनों डिसीज ग्रिप वर्क ओनली फॉर C6 एंड C7 पेशेंट, जिसको इंजरी C6 पे हुई या C7 पे, केवल वहां पे तीनों डिसीज ग्रिप काम करेगी। अगर C5 में है या C8 का पेशेंट है एट दैट दिस लेवल, वहां पे तीनों डिसीज ग्रिप होती नहीं है। अब तीनों डिसीज ग्रिप होती क्या? द अंडरस्टैंड दिस। If you ask the patient to do wrist flexion, then there will be opening of the fingers. And if you ask the patient to do wrist action, there will be closure of the fingers. How it works? Because there is a tension creating in the dorsal aspect. Due to that tension, after FDS, flexor digital superficialis, profundus, and palmaris longus, that will get closed. There is a tension created between this. Due to that, there will be closure of this. Now, यहाँ पे आपने स्लाइड में जो ऊपर फोटो देखोगे आप, if the patient is of C5 tetraplegia, and if you ask the patient to clasp the thing, patient will do like this. अगर आप पेशेंट को कहोगे कुछ चीज उठाओ, पेशेंट will clasp like this. क्यों ऐसा क्यों होता है? जो C5 का पेशेंट होता है उसकी इंजरी कहाँ पे होगी? हेड इंजरी, बिकॉज उसका क्या है? रिस्ट काम नहीं कर रहा, उसका हैंड मसल्स दे आर पैरालाइज्ड। पेशेंट कैन ब्रिंग हैंड टू द फेस, हाथ यहाँ तक तो ले आएगा मगर हैंड माउथ तक नहीं जाएगा। सो ही विल डू क्लास्पिंग लाइक दिस। मगर वही पेशेंट अगर C6 का है या C7 का है, वहां पे क्या होगा आपका? वहां पे आपका रिस्ट एक्सरसाइज वो थोड़ा सा वर्क करेगा और फिंगर थंब फ्लेक्सर का पैरालिसिस है। तो पेशेंट ने ब्रिंग हैंड टू द माउथ। जब पेशेंट हैंड टू द माउथ ले आया तो इसका मतलब उसका पिंच ग्रिप कैन वर्क। सेम चीज आपका C8 में होता है। C8 में क्या होता है? आपका इंटॉर्सिन, उसका थोड़ा सा पहले से हो गया और लंबी का काम कर दिया। नाउ नेक्स्ट थिंग इज दिस इज द एग्जैक्ट एक्सप्लेनेशन ऑफ तीनों डिसीज ग्रिप। आप अगर इफ यू सी द फर्स्ट पिक्चर, जब आपका रिस्ट फ्लेक्शन हो रहा है, आपका ओपनिंग हो रहा है फिंगर्स का, जैसे ही आपका एक्सटेंशन हुआ वहां पे क्लोजर हो गया। सो वी आर प्रमोटिंग पाल्मर ग्रिप and pinch grip. That is the main function of tenodesis grip in spinal cord injury, और हमें इसी पे काम करना है। हम लोग काम करते हैं। Now understand the concept. आजकल क्या हो रहा है? सर येलो बॉल खरीद लेते हैं, स्माइली बॉल, क्या हो रहा है उससे? What you are promoting? You are promoting the flexural synergy of the upper limb. क्या वो चीज सही है? पेशेंट पे का हाथ पहले फ्लेक्शन में जा रहा है, आप उसे और प्रमोट करो, कोई जरूरत नहीं उसकी। Rather than you work on the other things, sensory mapping और contactors, उस पे वर्क करो। सेम चीज आपका क्या होता है? अगर पेशेंट स्ट्रोक है, आपका स्पास्टिक हैंड हो गया और आपको फ्लेक्सर डोमिनेट कर लो, so there is no need of that. Now, अगर आपका टेनोडेसिस ग्रिप है, उसके दो तरीके हैं प्रमोट करने के। कैसे हम टेनोडेसिस ग्रिप प्रमोट कर सकते हैं? एक तो होता है FHS, फ्लेक्सर हिंज स्प्लिंट, that is very costly. Nobody, very few people can afford. दूसरा तरीका है this way. What is this? इसमें क्या किया गया है? टेपिंग, डॉर्सल एस्पेक्ट में टेपिंग प्रमोट कर दी और एट द एस्पेक्ट ऑफ फ्लेक्सर रेटिकुलम हमने वहां पे एक ब्रेस बना दिया। तो उससे क्या हो रहा है? रिस्क जो है आपका एक्सटेंशन में रहेगा, बट इसके भी ड्रॉबैक्स हैं। क्या ड्रॉबैक हो सकते हैं स्पाइनल कॉर्ड इंजरी में? इफ यू आर प्रमोटिंग tenodesis grip at the home and like this through taping, क्या होगा? It will lead to contactures, shortening of the head. So you cannot promote taping for tenodesis grip for the long hours. 3 घंटे 4 घंटे तो चलता है, बट बियॉन्ड दैट यू कैन नॉट डू बिकॉज उसके वजह से कॉन्टेक्स्ट को करना है, बट इट इज अ मैकेनिज्म यू कैन डू टू अवॉइड इनएक्सपेंसिव, जो कई पेशेंट अफोर्ड नहीं कर पाते एफएचएस, तो हम ये चीज प्रमोट कर सकते हैं। क्या एक्सरसाइज प्रमोट कर सकते हैं आप? यू कैन डू मोबिलाइजेशन स्ट्रेचिंग फॉर द तीनों डिसिप्लिन। ये है दिस इज द थिंग एफएचएस फ्लेक्सर हिंज स्पिन, जिससे हम तीनों डिसिप्लिन प्रमोट कर सकते हैं। इट वर्क थ्रू दिस। अनदर वे, आप पेशेंट को bring to the long sitting position, ask the patient to give a force downwards, डाउनवर्ड की तरफ फोर्स करो, मगर अगर पेशेंट टेट्रापीजिया है C5 लेवल है C6 पे, पेशेंट थोड़ी बहुत फोर्स डाउन करेगा, कुछ फोर्स आएगी थेरेपिस्ट की, थेरेपिस्ट फोर्स प्रोवाइड करेगा, थ्रू दिस पेशेंट कैन ओपन द हैंड फॉर ओपनिंग। नाउ अनदर टूल इज असिस्टिव टेक्नोलॉजी, वेरी इंपॉर्टेंट टॉपिक used nowadays in neuro rehabilitation. Assistive technology diagram. Assistive technology means कोई भी equipment, any equipment, it can be a piece of paper, it can be a piece of plastic, which can be used for product system and can innovate something for a patient welfare. जो भी चीज पेशेंट के वेलफेयर के लिए यूज हो रही है उसे हम असिस्टिव टेक्नोलॉजी कहते हैं। जब हम बात करते हैं लेवल में असिस्टिव टेक्नोलॉजी, लोग सोचते हैं अरे पता नहीं कितनी हाई-फाई चीज है, असल कुछ नहीं है। असिस्टिव टेक्नोलॉजी का मतलब कोई भी चीज जो आप पेशेंट के वेलफेयर के काम आ रही है, दैट कैन बी यूज्ड, ठीक है। बट आजकल असिस्टिव टेक्नोलॉजी के लिए हम लोग बहुत काम करते हैं, न्यूरो रिहैबिलिटेशन, मस्कुलोस्केलेटल, कार्डियोवैस्कुलर, वी वर्क अ लॉट फॉर दिस। नेक्स्ट, अब असिस्टिव टेक्नोलॉजी, ये मैंने एक डायग्राम बनाया व्हीलचेयर का, जस्ट आई हैव शोन द हाउ द व्हीलचेयर कैन बी यूज्ड फॉर असिस्टिव टेक्नोलॉजी। ये देखो, पेशेंट C5 लेवल का है, पेशेंट को मैनुअली व्हीलचेयर प्रॉपर करनी है, तो व्हाट यू विल पेशेंट विल डू, देयर विल बी सम प्रोजेक्शंस एट द लेवल ऑफ व्हील के हमने प्रोजेक्शन को लंबा कर दिया, तो पेशेंट उसको प्रॉपर कर सकते हैं आगे या पीछे। दैट इज असिस्टिव टेक्नोलॉजी एट द लेवल ऑफ C5 पेशेंट। नेक्स्ट C6, अगेन मैं C6 का आई हैव टोल्ड रिस्ट एक्सरसाइज, सो पेशेंट को पेज टर्न करने, पेज टर्नर, इट कैन बी यूज्ड एज अ असिस्टिव टेक्नोलॉजी। जो क्या करना है, मेक अ क्लैंपिंग थिंग, उसमें हाथ फंसा दिया और उसको टर्न कर दिया, तो इट कैन बी यूज्ड एज अ असिस्टिव टेक्नोलॉजी। नेक्स्ट थिंग, राइटिंग डिवाइस, पेशेंट हैज टू राइट, सिग्नेचर करते हैं, पेशेंट सिग्नेचर नहीं कर सकता क्योंकि पेशेंट को टेट्रापीजिया होगा, सो make a pen like that, it can be fit on the index finger, ring finger and patient can write with that assistive technology। देखने में चीज लगती है बहुत बड़ी, but it can be used in the home through low income substitute। Wrapping, entering the car, patient को रेस से सपोर्ट के लिए, it can be facility। just see it, just see combing the hairs, patient कॉम है, कॉम का जो पार्ट होता है वो छोटा सा होता है, उसको हमने बड़ा कर दिया। एंड व्हाट द पेशेंट डूइंग, हम पेशेंट को दो चीजें कर रहे हैं, एक तो पेशेंट अपना फॉलो भी कर पा रहे हैं और पेशेंट पाल्मर ग्रिप वो भी प्रमोट कर रहे हैं। सेम थिंग, इट कैन बी यूज्ड फॉर ईटिंग पर्पस, स्पैचुलास, कटलरीज, जस्ट सी वेरी, कोई इसमें कोई एक्सपेंसिव की जरूरत नहीं है एंड कैन बी यूज्ड। तो मैं आपको वो चीज बताना चाह रहा हूं, हेल्थ रिहैबिलिटेशन पे आप बहुत ज्यादा काम कर सकते हो थ्रू जस्ट लो इनकम। आपको कोई ज्यादा पैसा खर्च नहीं करना, जस्ट मेक सम टूल्स एंड यू कैन वर्क फॉर इयर्स टू कम। सेम थिंग, ये देखो, पेशेंट हैज टू रिमूव हिज गारमेंट्स, जस्ट मेक वेरी लॉन्ग रोड एंड पेशेंट ने स्वेटर पहना हुआ है, इट कैन बी रिमूव्ड, सेम इट कैन बी यूज्ड फॉर वियरिंग, वेरी इंपॉर्टेंट थिंग। फंक्शनल एक्टिविटीज, वी कैन प्रमोट द फंक्शनल एक्टिविटीज, upper extremity training, maximizing all these things। Same thing, just have a cup of tea, पेशेंट को कैसे कप पकड़ना है, that is the important thing। पेशेंट को कप पकड़ना नहीं आता, गिलास, पानी का गिलास पीना नहीं आता, so that can be used। Next thing, these are the things, how to squeeze the clothes, how to do this daily। आप दरवाजा बंद करते हो, खोलते हो, all these things that can be used as a tool, just so greatest challenge in treating the hand is to preserve the function in all their structures, not directly injured। आप वो फंक्शन प्रिजर्व करो. मैं वही कह रहा हूँ, जो उस पर्सन का फंक्शन उसको प्रिजर्व करो. डोंट वर्क ऑन द ऑल द इम्पेयरमेंट्स, केवल वो फंक्शन पेमेंट कर लो, पेशेंट अपना काम कर सकता है. मेन थिंग इज पेशेंट कैन अर्न हिज लाइवलीहुड. So rehabilitation of a tetraplegic into the community involves focused and dedicated care on the part of healthcare providers. This provides seeks to improve and maximize the quality and quantity of life. Effects of patient life is generally beneficial and lifelong. हमेशा beneficial होंगे और throughout the life it will work. So my home message is SCI is a relevant health issue because of the impact it has on the patient, and even though there is active research on the treatment development, being surgical or medical in order to achieve motor recovery. In the present time, there are only treatment to reduce the damage after SCI. केवल जो डैमेज हुआ उस पर हम काम करते हैं, हम उसके बेस लाइन पे नहीं जाते. तो वर्क एज अ मल्टीडिसिप्लिनरी केयर टू रिड्यूस द पॉसिबल कॉम्प्लिकेशन दैट मे अराइज. थैंक यू. Yeah, that is a very good question. Even I have not mentioned about that. Sir, what we are doing at our Jollygrant Hospital, when these things occur, we engage the patient in the daily activities through video gamings. We have a virtual reality apparatus in our equipment, and if the patient focuses during that, and patient practices through from easy tasks to a complex task through keyboards, through gaming purpose, so that the severity of that canvas it can reduce. Only thing is, I have mentioned one thing: practice. The more the patient will practice, the more the cortical reorganization will occur, and it will promote a pinch and palmar rate. So our main focus, हमारा main concern patient practice करे. Patient practice नहीं करता. जितना ज़्यादा practice करेगा, उसकी जो activities है और जो उसका side effects है, वो reduce होते रहेंगे, और उसकी जो focusing नहीं है, grasping, वो increase होता रहेगा. So that is the main purpose. That we are working in our hospital, yeah, and I have seen the improvements. Even sir, I was about to bring some. मैं अभी morning में बता था. I have devices something, some books sensory kit. तो sensory kit क्या है? That is I have made the paper जो use होता है greeting cards में. That is a thick paper. I have made that, and I have used that sensory kit as a different proprioception. First page उसका hard है, second page foam का है, third उसमें glass है. So that page can be used as a turner. Turner can be used as a or as a proprioception. So that is a very different tools. That's the thing.
I was just thinking of it because with the time now,
Yeah.
age is increasing.
Correct.
All parents, older people.
Yeah, yes.
Especially India, I'm talking about, of course, the whole their situation is there like that in Europe. Most of them are getting old, and there are no kids around Europe.
Yeah, definitely.
Where they should go? How will they be able to go to any profession? They won't be able to go. There is a lot of opportunity for
Yes.
kids.
Yes.
Start their nursery, old age nursery,
Correct.
nursery within that particular society. To go to any society, you start your own nursery. And you'll be earning more than 10 lakhs, so money is not a problem. Patient cannot go to the hospital.
Correct. That is the thing.
You have to serve them there itself. A lot of opportunities are there for the all physiotherapists in future. But yes, first you have to train yourself.
Correct.
All the new things, all the old things combined together will become. As you mentioned, he has given a lot of things that, yes, create your own way of.
Yeah, you have to be creative. You have to be creative. Yes.
Sir, I would like to acknowledge and express my gratitude to you. Can we have a big round of applause for sir? The whole world is looking towards one project today, the Western as well as the European countries, even in India. As the age is advancing, we are having more threat, and the projects which are going around every nook and corner is prevention of fall in elderlies. Now, how to stop that? Physiotherapist has a major role to play. What sir is talking about physiological damage. Now, a famous study, I just want to quote for students. In our world, respiratory world, we train respiratory sinus arrhythmias. When we inhale, heart rate will go up. When we exhale, heart rate will go down. So just by training the length, that is I/E ratio, in variety of combinations, we can suppress these physiological tremors, and these physiological tremors are reflected in locomotion. So the imbalance or disorders which are being reflected in the temporal and spatial parameters of gait can be reduced just by medical intervention as well. So I congratulate you, sir. It was a fantastic talk, and it was a very good message for students. Low cost technology to high cost technology, how to incorporate. It also incorporated the orthotic fabrications as well as engineering part, which is to be invited in physiotherapy profession more. And the best part, what I love for this talk was, hand functions are not only restricted to the field of neurophysiotherapy. Our central nervous system developed because we develop hand functions. So the upper thing goes to the sensory homunculus to the hand, if you see the reservation to it. So what all things we will do with the hand, that will get registered as in the memory trace and perception trace. So my teacher used to tell me when I was a student, Professor S. Balasubramaniam, he is one of the famous physiotherapists, and our country is proud of him to be a like Indian physiotherapist. He used to say कि जो भी skill आप patient को सिखाएंगे physiotherapy के नाम पर, neuro patients को especially, that is just a technique, and that technique का success depend करता है when you know how to transfer that technique into activity of routines, areas. So technique is that important, and the transfer of technique is also very important when we are working with neuro patients.
Correct.
Thank you so much.
Sure. एक चीज मैं ऐड करना चाहूँगा, अभी पल्लव सर ने एक चीज बोली है गेट के बारे में. मोस्ट ऑफ द स्टूडेंट्स जो यहाँ पे बैठे हुए हैं, उनको कहीं पर गेट की डेफिनेशन, व्हाट इज गेट, सब लोग ये कहते हैं सर स्टाइल ऑफ वॉकिंग, पैटर्न ऑफ वॉकिंग. एक्चुअली गेट होता क्या है? इट इज नॉट ओनली कंसर्न विद द लोअर लिम्ब, इट इज कंसर्न इवन विद योर थोरैक्स एंड अपर लिम्ब. एग्जैक्ट डेफिनेशन क्या है गेट की? इट इज अ स्मूथ रिदमिकल ऑल्टरनेटिव मूवमेंट ऑफ बोथ द लोअर लिम्ब along with upper limb and trunk. हम कभी भी बिल्कुल ऐसे नहीं चलते. जब भी हम चलते हैं हमारा आर्म स्विंगिंग होती है, आर्म स्विंगिंग, एंड दैट इज वेरी ऑब्जर्वेबल इन पार्किंसंस. कभी भी कोई भी पर्सन बगैर हाथ हिलाए नहीं चलता. इफ he or she is walking, that is awkward walking. चलते तो होंगे but that is awkward walking. तो आर्म स्विंगिंग इज कंसिडर्ड along with the pelvic movement and trunk. तो मैं मतलब मेरा मैसेज ये है, मैं कई जगह जाता हूँ एग्जाम देने, एज अ डिबेट, एज अ गेस्ट लेक्चर, सब लोग कहते हैं गेट इज कंसर्न विद द लोअर लिम्ब. नो, इट इज कंसर्न विद द अपर लिम्ब आल्सो. या इट इज कंसर्न विद योर हेड आल्सो. तो मेन मैसेज है आप लोग अपर लिम्ब पे भी काम करो, हैंड पे भी काम करो. दैट इज द मेन थिंग. थैंक यू.
And I won't take much time. एक last experiment करते हैं so that we can justify our presence in this conference, prestigious conference. All students, are you with me? We'll do an experiment. आप सब लोग अपना जो hand है उसको अपने knees पे रख लीजिए. Pronated. Both hands pronated on the knee. Close your eyes and try to inhale and observe your inhaling pattern. Breathing कीजिए और breathing को observe कीजिए. Observe it for 2 to 3 breaths. And here comes the magic. Now supinate your palm and again observe the same. Feel the difference. Yes? किसको यूरेका movement मिला? Fantastic. So what do you observe? When the palms are pronated, the depth of ventilation is less as compared to when the arm is supinated. Now this is just one simple example for you. Physiotherapists can create wonders.
Yeah.
So sir was talking of human gait. Human gait is all about ventral pressures of the human body, right? अगर arm swing नहीं है, suppose I am holding this mobile phone and I am walking, I am going for a morning walk. Definitely there will be more RSIs, repetitive strain injuries. Now so many studies are saying for the female population, those who are using long slippers, so they are walking with an altered gait. When we are walking and talking, our gait will change. The pattern of breathing will change. Everything will change. So gait has something to do more with the physiology rather than the hardware part of it. So very important, sir. Thank you so much. Congratulations.
Thank you so much, sir. I request the session chair and Professor Dr. Sahil Avrasa, Dr. Vishnuji, and Dr. Nihal Amrasa to please come up on stage to give a small token of appreciation to Professor Dr. Vaibhav.
I think a lot of patients have been shown by the students. Lucky for you all. In fact, you have listened very carefully. Most of the lectures, I'll request only 10 more minutes. You're getting more information from Dr. Keshav. He is from Australia and he will be guiding us on foreign aspects which you need to have a little bit break before you go for physiotherapy.
Thank you so much, sir. I'm very thankful for all the audience to be patient. So just, as I said, 10-15 minutes more, not more than that. So our next speaker for today is Dr. Keshav Raj Pondal, Senior Researcher, National Institute of Complementary Medicine, Western Sydney University, Australia. The topic for this presentation is Impact of Microplast Inhalation on Drug Health. I request sir to please address the camera.
Thank you, everyone. I'll skip a couple of my slides to make it quick. On academic background, I'm also a pharmacist. I did a master's and PhD in pharmacology, but today I'm covering a very different topic. One of the biggest environmental concerns of this year, and it's very relevant not only for the India but also global perspective, because the term pollution is not a new term for you guys in the major city like Delhi and nearby. One of the topic I'm covering today is plastic pollution and the impact of microplastic inhalation that is supported by the grant. Over the 7 years, I studied the impact of different types of environmental pollutants like bushfire smoke, urban particulate matters, the pollution coming from the industries, the smoke. In India, there is still a habit of biomass burning that releases a lot of toxic smoke we inhale every day. We studied this in different types of experimental models. We use cell lines, we use animals, we take cells from the patient sample, ex vivo culture, and we do those studies. Now, starting with the concept of microplastics, so we also know that these are very tiny plastic particles. They are less than 5 millimeters in size, and they are literally everywhere. They are in our food, drinks, any type of drinks, water, juice, beer, alcoholic drinks. They are in the processed food, especially if they are packaged in the plastic, they are in the textile, toothpaste, essentially everywhere. And also they are in the air. When they are in the air, we are breathing them every day. And it goes to the lungs and it has a lot of impact. So you may be wondering, on an average, how much plastic we consume every day or every week. So the scientists came up with this idea. They do the global average and they found that we get exposed to 5 grams of plastic every week. 5 grams is very easy to remember because it is equivalent to the ATM card, the bank card we carry in our wallet. That's exactly 5 grams. And this is because the exposure is to a lot of seafood. If we are wondering, if you are very fond of eating seafood, if you are vegetarian, you are not excluded from that, because still the vegetarian people, you consume the vegetable and fruits. You know the plastic that is dumped in the soil, break down and into the nanoplastic grains. And if there is a crack in the root, the plant can absorb those nanoplastic inside the fruits, and you consume that. So it's the problem for veg and non-veg, everyone consuming this plastic through the food. If we look at the statistic, 400 million tons of plastics are produced every year. And it's going to triplicate in 2026. The irony is, out of that 400 million, only 9% are recycled. The rest are dumped in the soil or in the water resource, or they are burned. And the statistic also says that 11 million tons, which is equivalent to dumping of 200 garbage truck full of plastic into the ocean, you see, every single day. And it is estimated that by 2050, there will be more plastic in the ocean than the fish. And that's a very serious issue. Now, this 5 gram equivalent of weekly consumption went viral with the many news agencies like WWF Australia, CNN, 9 News, Sydney Morning Herald, and scientists were previously concerned that the plastic is a problem for the aquatic animals because eventually they get dumped into the ocean. The fish or the sea animals, they consume that as a food. But when in this 2020 publication in One Earth, the scientists, what they did is they went to the Mount Everest, take a scoop of snow and analyze it. They found a lot of plastic in Mount Everest. So it's the problem from the sea level, the zero level, to the highest point, 8,848. So everyone living in lowland or in high mountain range, they are exposed to the microplastics. Now, if you exaggerate this condition, like 5 grams weekly, then in every month we are consuming 21 grams. In every 6 months we are consuming 125 grams. That's a ball full of plastic. Every year, a plate full of plastic. Every 10 years, 2.5 kilograms of plastic. And if you manage to live until 79 years, you'll consume 20 kilograms of plastic. The representative university is garbage bin, not just 1, 2, because one is 10 kilograms. Now you may be wondering, our body is like 60 or 70 kilo, where does this 20 kilograms goes inside our body? Well, they don't stay inside the body, it comes out with the poop or with the urine, but some of this plastic, nanoplastic or microplastic, they may accumulate in the body, organ, like liver, kidney, heart. And the time they travel, 24 hours or 48 hours before they come out, that's very crucial, they do a lot of damage. And I will show you that with a statistic. I'm just going to skip this slide. This is geographical distribution of where there is more plastic pollution. It's mostly on the southern part and the northern part in India. Skipping this as well. Now let's look at our food. The one I highlighted here with the underline is the data of the India. So you know, microplastic is found in 90% of the salt that is we derive from the sea. And the researchers, they found that in 1 kilogram of salt, sea salt, they found 700 particles. Another study is more scary, 1900 to 2300 particles in just 1 kilogram of salt. So we are consuming a lot. They might be less in other countries, but still those plastic, when it gets accumulated in our gut, when we take the salt, that does a lot of damage. Let's look at the behavior. So I was a bit surprised with this data from the Germany. We consider Germany as a bit of advanced. They have strict guidelines with the plastic regulation, but within one single-use plastic bottle like that one, 1 liter one, there was 2,857. In another study, there was even more, up to 10,000 microplastic we are consuming. So just one plastic bottle water. In the seafood, people, those who are fond of eating fish, we have bad news. This is the study of the China. They did the study in 27 different varieties, like species of the fish, and they found 17.2 particles in just 1 gram. The fish we consume is not 1 gram, it's 1 kilogram. So we need to multiply that number with thousands. That's 17,000 particles if we just eat 1 kilogram of fish. From India, there's a data from this particular species. It's a bit less than China, but still scary. And here's the representative image. This is the real image. That's the piece collected in 2008 from the North Pacific Guard, and they found 18 big pieces of this. These are the small pieces of plastic ingested by this fish. So if we eat fish, we are taking the plastic. Over 1 in 3 fish caught for the human consumption now have plastics. So if we are consuming a lot of these through drinks or inhalation of food, what does it do? What does the public studies tell us around? Well, it does a range of pathological events like oxidative stress, inflammation, senescence, is a premature aging, apoptosis, cell death, and it causes the disbalance of our good and bad bacteria in our gut, stomach, and intestine. Let's go through this figure here. The gut microbiota dysbiosis. What happens is in our body, when a baby is born, they have all good bacteria, 100%. But as we grow up, we eat unhealthy food, junk food, the number of good bacteria goes down, bad bacteria goes up. On top of that, if we are getting microplastic, it kills the good bacteria. So there's a disbalance of good bacteria. What it leads to do, there's a problem with food metabolism, digestion, a lot of other diseases. And when we are inhaling the microplastic, we are not just, it's not just plastic, it can contaminate with the pathogenic bacteria or virus, so it is a carrier for the pathogenic organism. It causes oxidative stress, it hampers or damages the immune cells that are essential to fight against the disease. It even causes carcinogenesis. Some of the recent studies is coming up that shows the cancer-causing potential of microplastic. It impairs the disruption of energy metabolism that it is done. It can cause neurotoxicity and reproductive toxicity. These are a couple of screenshots of the recent review article. We have highlighted the role of microplastic in lung disease and neurodegenerative disease. Now I'll go through my own data, okay, what I have found through the funded project so far. So in 2023, we got this triple-I microplastic grant, and we divided it into 3 years. In the first one, we do the inhalation studies. If you give the mice through the nose, what happens to the lungs? And we did this in 2 different settings: the acute study of 1 week, so 1 week intranasal, and then we did the chronic study of 8 weeks. So when we convert the mice age to human, we need to understand it this way. So 1 mice day is equivalent to 100 human days. So 1 week I'm exposing is equivalent to 2 years, almost 2 years in human. But 8 weeks in mice is equivalent to 15 years in human. That's exposure. And I did that in the healthy mice, but then I got interested what happens if I give the same microplastic in already pre-existing disease. So I can make the mice asthmatic, giving a house dose my disease allergen, and on top of that I give a microplastic to know if a human who has asthma gets microplastic, it will worsen the case. And then after I know the pathological event, I'm a pharmacist, I need to know the treatment. So I treated the mice with 4-octalate, which is a protein anti-inflammatory compound, and it's a very novel and promising compound these days. In another set of aim, what I did is oral gavage. I gave the same microplastic to the oral, and then I want to know what happens to that level of good bacteria and bad bacteria. And for the ex vivo, I take the cells from the patient healthy versus the COPD or asthma and expose the microplastic to the cells and assess the pathological difference in terms of cell viability, inflammation markers, oxidative stress, and sensors. So this is just a microscopic image of microplastic, and I was particularly interested in that size range, 0.2 to 9.9, and there is a reason for that. In terms of respiratory physiology, the inhalation, the particle size is very important. We don't inhale very big size, less than more than 10 micron is not relevant because they tend to settle in the upper respiratory tract, that is mouth and throat, so we don't get lungs localized damage. And I'm not interested in very ultra fine particles less than 0.1. We call it ultra fine or nanoplastic because 50% of the inhaled nanoplastic we exhale as we breathe in and breathe out, and the remaining 50%, once it reaches deep inside the lungs, it crosses the pulmonary barrier and it goes to the circulation. When it is in blood, it goes to any organs. And I'm not interested because I am checking only on lungs. So that's the reason I purchased this from the cost in California. And then the first study is 1 week. Let's look at the result. I have only 3 groups: the healthy control groups that receive saline solution, the microplastic at a low dose of 1 milligram per kilogram, microplastic at high dose. And I decided this dose based on the human equivalent exposure in highly polluted countries for the high dose and low polluted country for the low dose. So here is the result. When I flush the lungs of the mice with the fluid and collected the fluid back, I can collect the inflammatory cells in the lungs, and I can stand them and count them. When I count them, compared to the vehicle group, healthy control, with the 10 milligram per kilogram of microplastic, it gives a lot of total cell count, which is infiltration of the inflammatory cells. And the name of this inflammatory cells is macrophage, neutrophil, lymphocyte, and eosinophils. And that's the representative image. So these are individual cells. These are macrophages. A lot of macrophages here, a lot of macrophages in the 10 milligram per kilogram. And this is the enlarged image of single macrophage. You can see a clear nucleus here, the purple color, and these are the microplastics. What macrophage does is they love to eat, they engulf, they phagocytosize. They can phagocytosize anything, either it is bacteria or microplastic. They think it's a noxious, it's a pathogenic thing. And in another set of experiment, I was interested to look at the lung structural damage. So I did a couple of studies. The first one was hematoxylin and eosins to know if there are infiltration of these immune cells or inflammatory cells in the lung wall. We call it parenchyma. And while there was a trend, but I couldn't see significant difference. So 1 week of exposure is not enough to cause the structural damage. And same with the AD pass. What I'm looking here is, this is the airways of the mice. If there was positive staining, I could see the dark purple color mucus secreting cells called as goblet cells. Then I count the number of the goblet cells, but I couldn't see in the 1 week exposure. So still, it's not that scary that 1 week caused a lot of structural damage. The next staining is collagen. For the collagen division, we call it serous red and fast gains. Here we are looking at this red color staining around the airways. If there is a lot of collagen deposition, we consider the lungs is undergoing remodeling. And when there is a lot of collagen deposition, it affects the lungs' ability to breathe in and breathe out. Because lungs is like a sponge. It expands and collapses when we breathe in and breathe out. If there is a collagen deposition, elasticity will not be same and we can't breathe properly. But the good news is 1 week of exposure is not that bad that it can affect the collagen deposition. And then we can also measure the lung function. The way we breathe in and breathe out, we can do the same thing with the mice. We're using a machine called FlexiVent, and when we measure the different parameters, there was no statistically significant change here. Now, if I go for the chronic exposure, the same dose for not 1 week, for 8 weeks. Let's see how damaged it causes. But now it's going in the, up to like nearly 10-fold increase in the high dose. Everything goes up, like macrophages, neutrophils, lymphocytes, eosinophils. We can see a lot of these inflammatory cells here. These neutrophils was very less in 1 week model. Now we can see this. These are neutrophils, these are lymphocytes, macrophages, and small part here, these are lymphocytes. And if these inflammatory cells, which normally in the basal level or healthy level, these cells should flow in the blood circulation. They should not come into the lungs. But what is the marker, what is the signaling pathway that is causing those cells to come from blood to the lungs? I have an answer here. So when I check the bulb, the MCP1, which stands for monocyte chemoattractant protein, what it does is, if it is high in the lungs, it attracts the monocyte which is normally in the blood circulation to the lungs. That's why we are seeing a lot of macrophages or monocyte going up. The next chemokine is KC. That's the marker for the neutrophil chemoattractant that attract the neutrophil normally flowing in the blood towards the lungs. And these immune cells, when they come into the lungs, release a lot of inflammatory markers and do a lot of damage. A lot of other markers as well. I'll say interleukin-1 beta and interleukin-10 as well. But I couldn't see that same pattern in the serum. Why? Because the size range I chose was between 0.2 to 9.9. So those plastics are confined to the lungs. They didn't weigh to the circulation. So the same thing is only in the lungs fluid, but not in the serum. Now, in terms of collagen division, here we can see very clear difference. You need to know two things here. This is the airways, the white part. Look how narrow is this because of this inflammation. The epithelial here and here is like almost 3-fold increase. And look at this red color around the airways compared to this vehicle. It's a lot. That's a lot of collagen deposition. And what it does, it affects lungs elasticity. The lungs goes airway remodeling, and then we cannot breathe properly. The next, staining with the hematochelin using, we can see a lot of inflammatory cells now coming into the lungs parenchyma as well. And as I said earlier, like I was interested in these purple color, dark purple color cells here, represented by the arrowhead. We can see with respect to this vehicle, a lot of these goblet cells that produce the mucus. When there is a lot of mucus, the airways pipe gets narrowed and then they obstruct the breathing pattern. And finally, with the lung function, again, it was same result as 1 week and 8 weeks. We couldn't see any significant difference. For your reference, if I give the mice with the cigarette smoke, the tobacco cigarette smoke that I do with other model, we can see this emphysema a lot with the 8 weeks model, but not with the microplastic. Now, here's the summary. If we get exposed to 1 week of mice, which is 2 years in human, what it does is induce a mild inflammation in the lungs, but no structural change. Here, the only green one is the significant, red one is not significant. But if you get exposed to 15 years, I'm 40 years, I already get exposed to 15 years worth of plastic. So what's going in my lung? Here is the result. The inflammatory cells goes up. This is because of this key cytokines going up, and structural damage. I might not breathing as good as the newborn baby because my lungs has a lot of microplastic inhaled throughout like 20 years, past 20 years. So all this goes up. And we identified this with the time because 1 week and 8 weeks, and my recent PhD student is working on these two models. We have already finished with endpoint, and we have tested the potential of fluorocthalide to reverse the damage. And in another set, we expose the mice to the house dust mite to make allergic asthma, and on top of that, we are giving them microplastic to see if it does more damage. So these are the analysis ongoing, and we are planning for publication in future. If you want to learn more, there's a lot of literature, from the WHO. I would suggest you go to this website called Britannica.com. They have a very cool animated and cartoon figures, easy to understand in layman language. And there's another website called Surfer Against Sewage that has a lot of information about microplastics. I'd like to thanks for your attention, and these are all the people I'd like to thank for the funding body. Thanks.
Thank you so much, sir. Now I would request our session chair to please give a token of appreciation to Dr. Keshav Rajmangala.
Sir, can I ask one question, please? Sorry, thank you for giving nice presentation, Dr. Keshav. Actually, I have one question. Means you are using microplastics for 1 week inhale exposure to the animals, right? So there is one reason called olfactory epithelium where the blood-brain barrier is compromised, right? So when you inhale any microplastic in the micro range, so there is a chances that some of that particles may also enter into the brain. So that route. So if you give inhalation, right? So did you observe any significant changes in the behavior of those animals, or any inflammatory marker in the brain? Means because that also triggered the central, that microglial activation.
At the end, what we collect is lung frost, and then we are interested in liver and kidney. Only our collaboration will work on that, but we didn't collect it in the brain. One of the reasons is the size. We didn't give them nanoplastic or microplastic in the microwaves. And then we couldn't see it in the serum, since the microplastic is not yet in the surface. But one of the screenshots was my review article where we have to explain how microplastic of Alzheimer's and Parkinson's. That's because with the disrupted blood brain, it's easy to observe the brain and
because you showed that size range of your microplastic is within 1 micrometer, right? So there are some literature available which shows that if your particle is within 1 micrometer, so that may have chances to enter in the brain also, if you're giving inhalation.
Yes. If we could have like collaborated with investigating brain research, we could have definitely collected the brain research and provided, but we are on this study on the lungs, so that's all. But definitely that is one of the things we do.
Thank you.
You mentioned that throughout life we are consuming a lot of microplastic. Is there any way how to remove them from our system? And another thing, the next question is, which organ is most affected by
when they are in lungs, the lungs has the various roles of ciliary layer. These epithelial are ciliary. They have a cilia that moves to clear. But they can't clear effectively, and a lot of microplastic before the ciliary layers are engulfed by the macrophage. Macrophage eats. And this macrophage clears to the threshold and the drop. They literally die. When they die, they release a lot of inflammation markers. So that's very difficult process. And with the gut, when we ingest it, it's easy to come out in the food, but the time they pass through from the stomach intestine, they kill a lot of good bacteria. That's one of the things. Regarding how we can prevent it, just through water. If we do reverse osmosis, we can prevent. But boiling water is not a good solution. Boiling it kills the bacteria, but not microplastic. Plastic does out. And we talk about alternative, like for example, some of our here students take that. We have alternatives. We have a wooden chopping board. We go for that. If we chop the fruits or vegetables with a sharp knife, we might not see but small plastic leaves or in the cooking in that. Kettle, we can go for steel kettle or glass kettle to boil the water. If we are using a dryer to dry our clothes, one single dryer can release medium source microplastic in the environment. We are in our kids who are just newborn, the immune system in not that strong, so they are the ones which are at risk of losing pregnant, elder population. They are very,
How we can calculate how much it is?
So that's the global average where scientists don't agree. It's different controversy because it's not same level. Like India, it might be a lot of there is no very advanced technique yet, but most of these are sophisticated. Microsoft it can be easily to the blood. We can also do the food samples, like the food, easily quantify the number of there is a couple of areas that's of course because when we are in the sales, our sales can get stressed whether it's microplastic or any other cigarette smoker and then there is a reactive oxygen. And reactive oxygen is coming from microphone, they are more powerful than the kids. And that's the carcinogenic potential.
So somebody got a huge amount of plastic, right?
Yes.
So is there any technique to remove or you can take any preventive action for that?
From the body. That's the thing. We don't have that technique. That's why we need to improve our technique. We need to reuse, recycle or reduce consumption of the plastic.
Because the ultimate product of this contamination is the patient is getting infected with common disease, right? Cancer, diabetes, or any other which is chronic type of things. So is there any technique by which we can reduce the amount of infection or something like that? Infection or in the form of disease.
So the infection is linked with because they are a vector, they are a carrier for the microorganism. For infection, we go for the common modality like antibiotic therapy, but we need to target reducing the microorganisms. We can't dump the plastic in the soil and then clean it. Yeah. So yes, keep changing our own habits.
Thank you.
Please on stage to please give a token of appreciation to Dr. Keshav Rajkumar. Thank you so much, everyone. I request all the audience to proceed for the lunch. The student refractory. Please report back by 2:30 sharp.